It's a pleasure for me to present Dr. Cecilia Scorza, a longtime friend and an excellent scientist, for the next plenary lecture. Since the beginning of her career, Cecilia was interested in psychiatric disorders. She got her PhD in neuroscience at the University of the Republic in Uruguay in 2001, and did a postdoc at the Instituto de Investigaciones Biomédicas in Barcelona. Then she returned to Uruguay to build a career as an independent scientist, and since 2014, she is the head of the Department of Experimental Neuropharmacology at the Instituto de Investigaciones Biológicas Clemente Estable in Uruguay. During those years, she made important contributions, among others, in the field of the neurobiology of drugs of abuse. Especially, her research group has become a reference in the study of the neurochemical basis and the pharmacology of smokable cocaine. This is the research Cecilia is sharing with us today in her talk titled "A Decade of Research on Smoke Cocaine." So thank you, Cecilia, very much for being with us today. And I just want to remind the audience that all the questions, you can write them in the chat, and I will transfer to Cecilia. So thank you very much.
Well, good morning, everyone. First of all, I would like to thank the organizer of the meeting, especially Dr. Giselle Prunell, for giving me the opportunity to share with you our research on smoke cocaine during the last 10 years. This is the plan of the talk. I will start with some prevalence data of cocaine use and the different form of cocaine. I will mention some aspects of the phenomenon of smoke cocaine, which led us to involve in this investigation, our first questions, our published studies and results with a particular focus on some of them. And finally, I will present a brief summary of the ongoing experiments on two novel therapeutic strategies involving cannabinoids and gut microbiota. As you probably know, cocaine is the prototypical psychostimulant drug. Until now, it's an illicit drug of abuse, and it has a natural origin from erythroxylon coca bush. The last official report indicates that the prevalence data is 0.4% of adult population in the world has used cocaine in 2019. But cocaine use varies across regions. So in South America, the prevalence data estimate that 1% of the adult population use cocaine in the same year. But the relevance of this topic is associated with the cocaine use is associated with several health problems in the people who consume this drug, like, for example, addiction, of course, cognitive impairments, cardiovascular diseases, and a very high risk of consumption of this drug during pregnancy. Unfortunately, there are no approved medication for treat the psychostimulant use disorder. So this is an urgent topic for preclinical and clinical research. This scheme shows different forms of cocaine, coca paste or CP, cocaine hydrochloride, and crack. But there is two forms of cocaine: the hydrochloride salt, which is basically snorted or even or less intravenously injected, and cocaine base or free base, which is can only be smoked. So coca paste and crack, coca paste or CP and crack are the two smoke forms of cocaine, but there are some differences between them. Coca paste or CP is an intermediate product obtained in the process of the cocaine alkaloid extraction from the coca bush until the obtention of the cocaine hydrochloride. And for that reason, coca paste contains impurities, including other alkaloids from the plants and solvents or other compounds required for the chemical process. And we already know that CP is adulterated for se. On the other hand, crack is a posterior step after the cocaine hydrochloride. And crack is prepared specifically to transform the hydrochloride salt to cocaine base, which can be smoked like CP, like coca paste. While cocaine has been used for decades, crack emerged in the 80s, specifically in the USA, but CP is mainly consumed in South American countries like Argentina, Colombia, Chile, Peru, Paraguay, and Uruguay. And crack is almost exclusively consumed in Brazil. And sometimes there are reports that forensic report that there is no differentiation between crack and coca paste. Unfortunately, the spread use of coca paste started with a social and economical crisis in 2002 in Argentina and Uruguay, although there are few reports of CP consumption in other countries of South America from the 70s. There is a big problem with the people who consume CP and crack because they belong to a social, economically marginalized situation. So it is complicated, sometimes it's complicated to estimate the exactly number, or the real number of the people who consume this smoke cocaine. And another problem, a big problem, is the impact on the health of people who consume this kind of smoke form of cocaine, and the specific brain effects are not systematically reviewed or studied. When we began to study the brain effects of smoke cocaine, in the few publications that we found, there is a clinical consensus that propose that smoke cocaine induces a prototypical clinical profile very different from the cocaine hydrochloride, which is characterized by a high and fast dependence. And one of the main factors that were thought to explain this clinical profile was the route of administration. Several reports in the literature of drug abuse shows that the effect of different drug abuse could change depending on the velocity of reach the brain. So it has been well recognized that the route of administration is a key parameter to increase the vulnerability to addiction to drug abuse, like intravenous route of administration. Pulmonary inhalation is a fast route of administration compared with the oral or the intranasal or even the intraperitoneal way of administration in the case of preclinical studies. Without discarding the relevance of the route of administration, we thought that another factor that should take into account was the chemical composition. So based on that, on this phenomenon of smoke cocaine, we focused our research mainly in coca paste, and there were our first questions in the early step, in the first stage of the investigation. What factors can explain the abuse liability of CP? Is the pulmonary inhalation the main factor, or the presence of other substances like, for example, adulterants participate in the CP brain effects? Can we reproduce the pulmonary inhalation route in animal models? Well, we'll see how we address these issues. Firstly, we reached an agreement with the Technical Forensic Institute and the National drug both from Montevideo, Uruguay, to obtain CPCs samples from Uruguay. And until we started studying the pharmacological effects of CP samples, no forensic data were available to know the chemical composition of these CP samples. So we firstly focus our studies on the chemical composition of CP seed samples. I would like to mention or remark that these are samples that could be potentially smoked by drug users, giving us a realistic model to study the central the brain effects of coca paste. In this study, we identify and quantify the cocaine content and the content of different active adulterants in a very large series of coca paste disease samples. The results of this study also is available in this link, since this work was supported by the CICAD. CICAD is the Comisión Interamericana para el Control del Abuso de Drogas. And this work was done with the collaboration with other researchers from Argentina, Chile, Paraguay, Brazil, and Uruguay. And we found that in this series of CT samples, the 89% of the samples were adulterated. And this table showed the adulterants that we found in these CT samples. We found phenacetin, caffeine, aminopyrin, levamisole, lidocaine, and benzocaine. And phenacetin and caffeine were the most prevalent adulterants found in these CT samples. And this chemical profile was very similar to other forensic data reported, but for this sample of cocaine hydrochloride and even crack disease samples from Brazil. And being caffeine, the most used psychostimulant in the world, and the very well-known psychostimulant substance, we decided to study the role of the participation of caffeine in the coca paste pharmacological effects. But firstly, we looked for the relevance of the adulteration process in the literature of drug abuse, and we found that the adulteration is a process that implies the addition of pharmacological active substances to mimic or to boost or enhance the effect of the main drug. In this case, the main drug is cocaine. In addition, the pharmacological interaction between the different compounds present in drug of abuse is commonly underestimated in the literature, and in the real effect of the drug of abuse in people who consume this kind of drugs. And of course, no data were available about the chemical composition of CT samples prior to our research. Interestingly, while caffeine and cocaine have different mechanisms of action, both substances share the same brain substrate. So it is very well known in the literature that the psychostimulant effect of cocaine and the rewarding effect of cocaine is mainly exerted by a dopamine uptake blockage by cocaine. And the psychostimulant effect of caffeine is mainly induced by the antagonism of the A1 and A2 adenosine receptor. So both mechanisms lastly induce an increase in dopamine neurotransmission in the mesolimbic circuits and the dorsal striatum regions. That is the base of the psychostimulant and the rewarding effects of the drug of abuse. So in 2011, we published the first international paper work that we found in this paper. We found and we demonstrate that caffeine was able to enhance the acute stimulant effect induced by different CP samples with different proportion in cocaine and caffeine content. And also, we established in this paper, we established the concept of the CP surrogate, which is the combination of cocaine and caffeine, and at a specific proportion found in CP samples. And we established this concept since the impurities of the CP samples did not participate in the stimulant effect, in the acute stimulant effect induced by different CP samples with different proportion of cocaine and caffeine. And so we wonder if caffeine was able to enhance the CP acute stimulant effect using the real route of administration through the pulmonary inhalation route in animals. But firstly, before that, we needed to test the integrity, the chemical integrity of cocaine and caffeine after volatilization, after heating. To do this, we replicate the method used by the CP consumers. The consumers use these devices to consume, to smoke coca paste or even crack. And this is in our model. This is a stainless steel tube connected to a syringe, and CP, a specific amount of CP is placed in this part of the tube, and on the other end, the syringe is placed, and a lighter was turned on, and the CP was heated until the volatilization. The smoke of the volatilization of the different CPs is get here in the syringe because the syringe is moved away, and the smoke contained in this syringe is after placed in a Hamilton syringe. Is a very small Hamilton, it's very small syringe, which is injected. The smoke is injected directly to a GZMS equipment. This is the representative chromatogram of GZMS of the smoke of a CP, adulterated CP sample. This is cocaine, caffeine, and phenacetin. As you can see, the integrity of this compound is preserved, and these other compounds appear when cocaine is volatilized. Specifically, this compound AEME is the prototypical biomarker of a smoke form of cocaine, CP or crack. So we confirm the integrity of caffeine, injecting the smoke of a standard solution of caffeine, and we can see that no combustion products were observed after the volatilization of caffeine. So we confirm the preservation of the integrity, the chemical integrity of caffeine after heating. So taking into account that caffeine and cocaine were preserved after volatilization, we tested the role, the participation of caffeine in the acute stimulant effect induced by different CP samples with different content of cocaine and caffeine. We selected these samples, CP1, CP2, CP3, and CP4, and CP1, CP2, and CP4 contain a high content of cocaine, and CP1 and CP3 contain a higher content of caffeine compared with CP2 and CP4. So we using the same method described before, we volatilized these different CP samples, and after that, animals were passively exposed to smoke of these different CPs in this inhalation chamber. Animals spent here for 10 minutes, and after that, animals were placed in an open field paradigms, and the distant move or the locomotor activity was recorded using the video tracking software. So we can see the results. This graph are the distant moved induced by CP1, CP2, and CP3. We can see that this is the composition, the chemical composition of each CP sample. As you can see, CP1 induced a significant effect in the locomotor activity of the animal compared with the control group. Animal treated with CP2 behaves similarly to control group, and CP3 induced a significant locomotor activity, a significant increase in the locomotor activity of animals. Interestingly, this effect was similar to the CP1, with having a half content of cocaine and a higher content of caffeine. And the following question was if coca paste can increase dopamine release in the nucleus accumbens shell, which is a common property of drug of abuse and underlies the rewarding effects of the drug of abuse. This is a very well-known, recognized effect, and this parameter is commonly tested when you are studying a drug of abuse. So the dopamine release is provided by dopamine neurons from the VGA projecting to the nucleus accumbens, describing the mesolimbic system and underlying the rewarding effect of drug of abuse. So using the same method, again, the same method described before, we designed a series of macrodialysis experiments in freely moving animals and permanently perfused animals. So during the volatilization of CP sample, animals spent here in the inhalation chamber during 10 minutes, and after that, animals were placed in a dialysis or macrodialysis chamber to recover the dopamine extracellular levels, which were analyzed by an HPLC with electrochemical detection. So in this graph, you can see the dopamine extracellular levels in the nucleus accumbens shell induced by CP1 and CP4. This is the content of cocaine and caffeine for the two CP samples. And we can see that CP1 induces a significant increment in the dopamine levels, while the effect of CP4 was less pronounced and short, long-lasting. And so for us, these experiments or these results are very, very, very important, because as far as we know, this is the first demonstration of a smoke form of cocaine with or without caffeine inducing an increase in dopamine extracellular levels in the nucleus accumbens shell. So this finding, and in addition to the previous one, the behavioral effects using the real route of administration, suggest that the presence of caffeine in CP samples may potentiate the stimulant effect and the rewarding effect, and lastly, the abuse liability of CP. Well, from the continuous study, the pharmacological interaction between cocaine and caffeine in specific proportion found in CP sample, in different CP samples. But for reason of time, I will show you briefly a series of behavioral studies and results from behavioral studies and their molecular basis, which led us to propose the studies about two different therapeutic strategies that I will present in a while. In this first study, we confirmed that caffeine can enhance the reinforcing effects of cocaine and also the motivational value of cocaine in a CP surrogate administration. We inject animals with a specific proportion of cocaine and caffeine found in a specific CP sample, and we use the self-administration paradigm. So we found that the animals treated with a combination of cocaine plus caffeine have a significant high breaking point compared with the cocaine treated animals. Remember that this the breaking point is calculated from a progressive ratio as said in the protocol of self-administration paradigm, where animals have to poke more times to obtain a dose of the drug, and the effort of animals willing to make to obtain the drug is calculated through this parameter. So after that, in this paper, we demonstrate that the presence of caffeine can enhance and facilitate or accelerate the expression of the sensitization induced by different coca-based samples. And we know, as you probably know, this phenomenon, the sensitization, the behavioral sensitization model, the craving and relapse induced by psychostimulant drug of abuse, and this phenomenon is related with a progressive or is observed as a progressive and permanent increment in the locomotor activity of the animals exposed to repeatedly exposed to a psychostimulant, a withdrawal period, and the re-exposition of the psychostimulant. So there it has been well recognized and well described some neuroadaptations in a reward system, underlying this sensitization phenomenon. And in this study, we further investigate the molecular basis of the molecular changes associated with the locomotor sensitization induced by different coca-based, adulterated coca-based and non-adulterated CP samples. So again, we demonstrate that caffeine collaborates in the induction of molecular changes underlying the CP sensitization. And I would like to remark one of these changes that associated with the RNA expression levels of CNR1 is the gene of the CB1 receptor. So we observe an increment in these expression levels of the gene in an adulterated CP sample compared with a non-adulterated CP sample. So this parameter suggests the participation of the endocannabinoid system in this in the expression of the sensitization induced by CP sample. And finally, in this study, we demonstrate that caffeine can enhance the reward-associated memory measured in using the conditional place preference in rats. And this effect was associated with specific changes in immediately early gene expression. So to summarize the result presented so far, we can say that as the combination of cocaine and caffeine in a specific proportion found in some CP samples, this combination participates in the stimulant effect, in the acute stimulant effect, in the rewarding and motivational effect of CP, in the sensitization, in the locomotor sensitization phenomenon, and the reward-associated learning. So we proposed with this, all these results, we propose in all these papers, we propose that this pharmacological interaction between cocaine and caffeine can contribute to the high and the fast dependence of CP consumers. And in addition, our results highlight the relevance of the active adulterants in the brain effect induced by coca paste. And now I will present briefly the first stages of two novel approaches that we are studying for the treatment of psychostimulant use disorder. One of them is based on the study of cannabinoids, especially focused on cannabidiol. Cannabidiol, as you probably know, is a non-psychotomimetic compound of cannabis without rewarding properties and a high degree of safety. In the last year, a lot of preclinical studies and some clinical studies suggest that cannabidiol can be used for cocaine use disorders. A lot of studies, preclinical studies, have shown a vast therapeutic properties of CBD, like anxiolytic, antipsychotic, antidepressant, anti-inflammatory, anticonvulsivant properties for cannabidiol. And all these properties led to propose cannabidiol as a possible treatment for psychostimulant use disorder. These two excellent reviews summarize data about the advantages and the putative mechanism of action of cannabidiol in the for the treatment of psychostimulant or substance use disorders. And in following this direction, we studied the effect of cannabidiol in the expression of the sensitization, the locomotor sensitization induced by the combination of cocaine and caffeine in a specific proportion found in CP samples. And we treat animals with cannabidiol and the combination of cocaine plus caffeine during 3 days, and after 5 days of withdrawal period, all animals on day 9, all animals receive a challenge dose of cocaine plus caffeine, and after that the behavioral study were carried out. And as you can see in this graph, this is the acquisition phase of the sensitization. The locomotor sensitization is before the withdrawal period. As you can see that animals treated with cocaine plus caffeine induces a very significant effect and a significant increase in the locomotor activity, since they want to the third day, and the pre-treatment with cannabidiol did not attenuate the stimulant effect induced by the combination of cocaine plus caffeine. In contrast, the pre-treatment with CBD, with cannabidiol, did attenuate the expression of the sensitization induced by cocaine plus caffeine. This is very relevant observation because this suggests relevant differences in those neurobiological processes that CBD is preserved. Our data agree with other reports of the literature which demonstrate that CBD can prevent neuroadaptations induced by the repeated treatment of cocaine, only cocaine, and without preventing the rewarding effect of cocaine that is associated with the acquisition phase. So while we have not studied yet the mechanism of action of CBD in this in the blockage or the attenuation of the expression of the sensitization, there are several targets involving the mechanism of action of CBD in these two reviews that I mentioned before that suggest us to interpret this our results, but some actions proposed to CBD are focused on the dopaminergic, endocannabinoid, serotonergic, and adenosinergic system. But there is one of particular interest to our study that implies the blockage of this protein, the equilibrative nucleotide transporter 1, that is the primary mechanism of adenosine reuptake. CBD blocks the ENT1, and when blocked after the blockage of this ENT1 protein, CBD induces an increase in adenosine levels in the brain, while this action in the literature, this action was associated with the anti-inflammatory properties of CBD proposed for this author. We cannot discard that this brain site can explain, at least in part, our data of the expression of the sensitization. We have a hypothesis, our current hypothesis is based on this idea. We think that there is a competitive action between the increased levels of adenosine evoked by CBD and caffeine for the antagonism of the adenosine receptor A1 and A2A. So this competition may result in an attenuation of caffeine action induced by CBD. We are working in this hypothesis in this moment, so I still don't have any results to show you. So maybe in the next edition of FALAN, I can share some results about this. The second therapeutic strategy that we are investigating is based on the modulation of the gut microbiota and probiotics. In the last years, very few last years, there has been a series of papers connecting the gut microbiota and psychoactive drugs. So, very briefly, the concept is this paper works in a concept that a modulation of the gut microbiota can improve some negative aspect or some pathological aspect of several mental diseases like depression, schizophrenia, autism, neurodegenerative diseases, and also some anxiety symptoms. So this author proposed that maybe the modulation of the gut microbiota can improve some negative aspects related with the drug addiction. And so following this direction, we had the opportunity to investigate the effects of smoke cocaine on the alterations in the gut microbiota of rats and exposed to cocaine, volatilized cocaine, and also active adulterants like caffeine and phenacetin. So in this study, because of time, I will present only, I will focus out the results only for cocaine. So as you can see in this graph, cocaine, animals treated with cocaine have a significant increment in the locomotor activity during 14 days from the day 1 to the day 14. So associated with this behavioral changes, we can observe that the abundance of the bacterial community of animal treated with cocaine tend to be very different from the rest, from the control group and the other substances, and in the same direction, the diversity of bacterial community was significantly lower in those animals treated with cocaine. And in this table, you can see the different alpha diversity indexes. And for example, Shannon indexes indicate that cocaine induces a significant lower in the diversity of the gut microbiota, and on the other hand, the dominance index shows an increment in the gut microbiota of animals treated with cocaine. So we, based on the concept that we can modulate the microbiota and modify some aspect of this the gut microbiota induced by different drug abuse. We wonder if the pretreatment with probiotic can prevent the effect on behavior, on cocaine behavior, and the effect induced on the gut microbiota, and see what happened. Unfortunately, there are I don't have any definitive results yet, because these experiments are being performed at this moment. So again, maybe in the next edition of the FALAN. To summarize, to finish, I will summarize the last part of the talk. Our results and the other reports in the literature strongly support a very promising role of cannabidiol in the psychostimulant use disorder treatment. But of course, more preclinical studies are needed to identify the mechanism of action of cannabidiol, and of course, clinical trials are more needed. For the last part of the talk, we and other authors proposed that the probiotic treatment may be useful in disorders of drug addiction, maybe as a monotherapy or in addition to some behavioral or pharmacological intervention. But there is a very important thing that it's very important to know which probiotic or the combination of which probiotic could be more efficacious in this topic. And this is a very, very new discipline that should be developed only if basic and clinical researchers generate a very close nexus between two disciplines, macrobiology and neuroscience, field. So to finish, I would like to really thank the people who took part of this research at the institute, in my lab, and in other departments of the institute and the university, and international collaboration that we have. And of course, CBD is donated by Phytoplan Research. And of course, I would like to thank the financial support to this research. Thank you very much for your attention.
Thank you, Cecilia, very much for this excellent conference, and congratulations to you and your research group for this excellent work. We have a lot of positive comments and many questions. We will go through questions. So Eduardo Orrego Escobar asked, do you think that research about cocaine therapeutic potential use could drive to exigence for its legalization, like was cannabis?
Well, thank you very much for the comments and the questions. I think, Eduardo, that for example, here in Uruguay, legalization of cannabis allows to regulate the recreational use of cannabis, and consequently, the medicinal use of cannabis emerged, and today is very well recognized the use of cannabis or cannabinoids to treat or some negative aspect related with cocaine use or cocaine addiction. But specifically, our results shows that the adulterants can impair some negative aspect of cocaine. So I think that harm reduction strategy can be or could be analyzed the adulterants and put in alert the clinician about the participation of the different things like caffeine, different things or negative things related with this kind of substance. So I think that the therapeutic use of cannabis in the cocaine addiction is a very good strategy to follow.
Okay, next question from Susana Castro Obregon. What about the behavior of animals inhaling only caffeine?
Okay, yes, well, for reason of time, I couldn't show you the effect of caffeine per se, but we did this experiment. We studied the effect, the behavioral effects of caffeine, smoke caffeine, the passively exposed animal to volatilized caffeine, and we observe a dose response increment in the locomotor activity induced by volatilized caffeine, but the proportion on the dose or the amount of caffeine used in this particular experiment are different from the composition of some CP samples, which have some specific proportion between cocaine and caffeine.
Okay, Francisco Estrada Rojo wants to know if the position of cocaine first, and then caffeine has the same effect.
Well, I really don't know the answer of this question. It's a experiment that probably should have to perform, because in our experiment, we exposed at the same time the cocaine and the caffeine. The interaction, the pharmacological interaction happens in the same moment. So probably when you pre-treat animals with cocaine and challenged animal with a caffeine injection or inhalation, I don't know what will happen. You have to do this experiment.
Okay, Alicia de la Fuente. Sorry, we have another question first. Mailident wants to know about the microdialysis with caffeine alone. Does she miss that?
Okay, well, the same answer. For reason of time, I select the studies to show the interaction between cocaine and caffeine, and leave the experiments of caffeine alone to make, maybe you can read the paper where I demonstrate the dopamine release induced by caffeine, volatilized caffeine. So we in this study we demonstrate that caffeine by a fast route of administration, the pulmonary inhalation, is able to increase the dopamine release. So in some way we demonstrate that one substance like caffeine or even cocaine have different effects or different responses depending on the route of administration that we use to study this effect. So caffeine is able to induce the behavior like a drug of abuse when you use the fast route of administration.
Alicia de la Fuente wants to know if the profile of the adulterants in sniff cocaine is different from the ones that you saw in CP samples.
Well, our studies about the chemical composition were about only for CP samples, not for cocaine hydrochloride samples. But I think I know from the Technical Forensic Institute, the analysis from the Technical Forensic Institute here in Uruguay, cocaine contains also some adulterants and contains caffeine as a main adulterant. So I don't know because we didn't perform this analysis, but I think that hydrochloride cocaine seed samples probably contain adulterants like CP.
Next question from Maryland. Will you experiment in the future if caffeine shows the habituation that Chiara saw in the prefrontal cortex with drugs and didn't with natural rewards?
Well, we didn't perform this experiment. I think that is a very good idea to follow. Yes, I have to study this paper of Chiara to design an experiment like this.
Maria Bermudez wants to know, do you know if the chew cocaine, like it is made by native people of South America, has some effects that could be compared with smoke cocaine?
Well, it's a very, very good question. I think that the route of administration can make the difference in the effect of cocaine or other substance of abuse. So I think that of course the effect of cocaine depends on the frequency, the amount of consumption. The route of administration is very, very important. So maybe the oral cocaine, like a tea of cocaine, or they chew with cocaine, can reproduce some negative effect of cocaine, but during a long time of consumption in this kind of in this way, like chewed or oral consumption.
Next question is from Eduardo Rego Escobar. He wants to know if there is any study about the rate of coffee consumption between consumers of drugs or addicted.
Sorry, Giselle, can you repeat the
yes.
question?
If there is any study about the rate of coffee consumption between consumers, consumer drugs or addicted.
I can understand the question. The rate of coffee.
The rate of coffee consumption.
Coffee consumption. Coffee consumption in cocaine addicted persons.
Not cocaine, addicted persons or drug consumers in general.
I don't know. It's a very, very good question. I don't know if the dependent person consume a lot of caffeine. Yes, coffee or caffeine. I don't know. Maybe there is a cross sensitization between the caffeine from coffee and cocaine, but I don't know. I really don't know.
Okay, next question is from Elaine Aparecida de Belmus Guimaraes. She says, excellent conferencia, Cecilia. Please, could you comment your results with antibiotics?
Well, yes, antibiotics. Well, Elaine, we didn't use antibiotic. We only used the exposition of cocaine, and we observed an effect, a very significant effect on the gut microbiota of animals exposed to volatilized cocaine, like an antibiotic. There is a paper, Kirali and collaborators, that use a pretreatment, a very long pretreatment with antibiotics, and after that they tested the effect, the central effect of cocaine in two paradigms, in CPP paradigms, place preference, and in the sensitization, the locomotor sensitization. And they found that the cocaine effect in these two paradigms increased the negative effect. With the in the animals treated with antibiotics. So they established a very close relationship between the gut microbiota and the central effect of cocaine. And we did, in this same direction, we studied the effect of cocaine in the gut microbiota of animals, but per se, without an antibiotic pre-treatment.
Next question is from Gonzalo Olivares. He asked, could it be that the changes in gut microbiota are due to changes in feeding habits due to drug consume or changes in metabolism in the host?
Sorry, can you take again, repeat
yes.
me again, Giselle, please.
Could it be that changes in gut microbiota are due to changes in feeding habits due to drug consume or changes in metabolism in the host?
I think that it's a bidirectional effect. When you alter the gut microbiota, you can alter the behavior of the person. So when we consume some drugs that alter the gut microbiota, you can impair or modify the effect of the final effect of the substance, like cocaine. So it's a bidirectional effect. We don't know if, which is the first change, the gut microbiota or the central effect, but it's a bidirectional situation. Okay. I don't know if I answered the question, because I couldn't understand the question deeply. I don't know how, where is the question? Maybe I can, could be the changes in gut microbiota are due to changes in feeding. Feeding habit. I understand the feeling. So sorry. The changes in gut microbiota are due to changes in feeding habit due to drug consumption. Well, yes. The gut microbiota is changing with diet. This is the first demonstration that the diet, the feeding habits can modulate the gut microbiota of person. So in addition, you can add or you can modify also the gut microbiota with the consumption of a drug. But our results or the preclinical results, you can test it or study the only one variable, like the effect of the drug of cocaine, without changing other variables, like, for example, the feeding of the animal. So in some way, the preclinical study helped us to understand which variable is more important to understand this bidirectional axis, no? And finally the effect of the drug abuse.
Okay, next question is from Juan Jose Lopez. He wants to know which is the effect of caffeine and cocaine on blood pressure and stroke risk. Drinking speed could potentiate the risk of cocaine consumers.
Well, I don't know, because we are not dedicated to a cardiovascular effect of cocaine, but yes, I think that the speed, drinking speed with a lot of amount of caffeine can potentiate the risk of the cardiovascular effect of cocaine, and also the addictive property of cocaine. Yes, I think it's a very complicated thing.
Okay, and our last question is from Liliana Marina Cancela. She asked, what about the impact of adulterants in the long-term effect of cocaine? And do you think that other structures could be implied in this potentiation of caffeine and cocaine-induced effects?
Well, thank you, Liliana. Well, the long-term effect of cocaine, we studied the sensitization phenomenon of induced by cocaine in presence of caffeine, and we observe neural plasticity events that are commonly reported by cocaine only, the use of cocaine. But I think that the adulterant can, like caffeine, can improve and enhance and facilitate this effect, this long-term effect of cocaine. And even when you use a fast route of administration. And do you think that the other structure could be implied? Yes. Well, for reason of time, again, I couldn't show you the study where we evaluate the metabolic changes in prefrontal cortex and nucleus accumbens, shell and core, and we observe metabolic changes, a very strong metabolic effects on these two regions related with the mesocortical and mesolimbic system, related with the rewarding effect of drug of abuse. So I think that all the structure belong the motivational and rewarding effect are involved in the effects of cocaine and caffeine. Yes, I think yes.
Okay, we are finishing this part. Thank you, Cecilia, very much. We have a short break and then we continue with symposiums in room A and room B. Thank you everyone for attending this part of the meeting.
Okay, thank you very much. Bye.
So welcome everyone. My name is Patricia Cassina from Uruguay, and I'm honored to chair the next symposium. First of all, I'm going to make an announcement. We are going to send you by chat a survey to complete some interrogations about the meeting. Please fill it up because it is important for us to see how many people are going with this Congress. So now, yes, we are going to start this symposium that is on glial cells in health and disease. These cells for many years were considered simply support for neurons, but now they are increasingly becoming important players in neuron function and development. So we have 4 outstanding speakers to speak about this issue. I'm going to present the first speaker. First speaker is Dr. Lorena Rela from Argentina. She is a biologist, graduated from the School of Science, University of Buenos Aires. Then she obtained a PhD with Lidia Supac at the Physiology, Molecular Biology and Neuroscience Institute of Buenos Aires. She was postdoctoral fellow at Yale University School of Medicine in USA, working on repairing mechanisms in olfactory system with Charles Greer. Her current position is Professor of Neurophysiology at the Department of Physiology and Biophysics, Facultad de Medicina, Universidad de Buenos Aires. And in addition, she is researcher at the Institute of Physiology and Biophysics, Bernardo Jose. The focus of her research is to understand the role of glial signaling in determining the regenerative capacity of neural circuits and maladaptive plasticity in neurodegenerative disease. The title of her talk is In Search of Modulators of Neuroplasticity and Repair. Thank you, Lorena, for accepting.
Hello, everyone. I would like to thank the organizers of FALAN Network in 2021 for giving me the opportunity to show our work. And I also would like to thank the audience for joining this symposium. Because this is a networking event, I will show a mix of published data, new preliminary data, and a little bit about starting projects. The main goal of our group is to identify and understand mechanisms involved in plasticity and repair of the nervous system. As you know, upon neural damage, there is loss of functions depending on which structures are affected. Regeneration of damaged tracts can occur, especially of peripheral nerves. However, the central nervous system is poorly permissive for regeneration, and we are interested in finding ways to improve its regenerative capacity. We use the mouse olfactory system as a model. Here, olfactory sensory neurons with cell bodies located in the olfactory epithelium lining the nasal cavity project axons to the first relay of information, the olfactory bulb in the central nervous system. These axons constitute the olfactory nerve. The olfactory nerve is atypical in that it is permissive to sustain axon growth throughout adult life, as sensory neurons turn over and are replaced by new ones. The olfactory nerve also regenerates and reinnervates the central nervous system efficiently after damage. This nerve is populated by specialized glial cells called olfactory ensheathing cells that have a mixed phenotype, sharing characteristics with Schwann cells and astrocytes, and are depicted here in red. They are present along the nerve from the lamina propria of the olfactory epithelium to the outmost layers of the olfactory bulb, and have been proposed as important contributors to the permissiveness of the nerve for axon growth. It was shown that ensheathing glia have neurotrophic properties both in culture systems and when transplanted to sites of damage, in particular to the spinal cord. The efficacy of ensheathing glia transplants is variable, and there is a need to understand which mechanisms operate to explain axon growth promotion physiologically in vivo. Here, I will tell you how we approach this problem and where are we now. Ensheathing cells have been intensely studied in culture, where they can establish connections throughout gap junctions, a type of intercellular connection mediated by connexin channels, permeable to metabolites and ions. We confirm this interaction with an electrophysiological approach in a more intact preparation. In olfactory bulb slices conserving the terminal portion of the olfactory nerve, we show that a dye introduced in an ensheathing cell through a recording pipette can pass to neighboring cells, which is taken as evidence for the existence of gap junctions. And in double recordings, we showed that when stimulating one ensheathing cell with a series of voltage pulses, current responses can be evoked in the stimulated cell, and responses can be also evoked in a neighboring dye-coupled cell, which is direct evidence of gap junction communication. Both dye and electrical coupling can be blocked with meclofenamic acid, a pharmacological inhibitor of connexin proteins, which constitute gap junctions. Here you can see that responses in the stimulated cell are reduced in amplitude, and responses in the coupled cell disappeared. Gap junction uncoupling by meclofenamic acid correlates with a reduction in membrane conductance, which is the slope of the current versus voltage curve. When analyzing the spatial distribution of electrophysiological properties, we observed that ensheathing cells located nearest to the olfactory bulb surface were more likely to be uncoupled than deeper cells, and were less sensitive to connexin blockers in electrophysiological experiments. And this correlated with a spatial gradient in the expression of connexin 43, the most abundant connexin in the olfactory nerve. To determine the contribution of connexin 43 in the formation of ensheathing glia networks, we used genetically modified mice to abolish its expression in ensheathing cells. We achieved this by crossing two mouse lines to obtain mice with two genetic modifications in the second generation offspring. One modification is the expression of a tamoxifen-inducible recombinase and the regulatory sequences of the proteolipid protein gene expressed by ensheathing glia. The other modification is the presence of LOXP sequences flanking a large part of the coding sequence for connexin 43. Mice carrying the two modifications undergo genetic recombination and deletion of connexin 43 coding sequences upon the administration of tamoxifen in the cells that express the recombinase, which in the olfactory nerve are the ensheathing cells. Recombination is not expected to occur in other cell types that do not express the recombinase. Here you see immunostaining against connexin 43 in the olfactory bulb, where you see that the terminal part of the olfactory nerve is rich in the punctate label characteristic of connexins forming gap junctions. This is a control tamoxifen-treated mouse carrying only the LOXP sequences. The punctate label disappears in a mouse also expressing the recombinase, but does not disappear in a vesicle-treated mouse. The electrophysiological recordings show that in control mice, the profiles were characteristic of coupled cells with large conductance that was reduced with connexin blockers, as you can see here as a change in slope in the current versus voltage curve. In contrast, mice with conditional deletion of connexin 43 showed reduced conductance with little sensitivity to connexin blockers. Here you see that the fraction of the membrane current that is sensitive to the connexin blocker is dramatically reduced in conditional knockout mice. And the pattern is similar both in adult and juvenile mice. Decoupling observed in control cells was also abolished in connexin 43 deleted cells. We then asked whether the expression of connexin 43 by enchything cells played a role in the permissiveness of the olfactory nerve for olfactory sensory neuron turnover and regeneration. And I will show you some preliminary data. Both during normal turnover and after damage, new sensory neurons are derived from progenitor cells at the base of the olfactory epithelium. They give rise to immature sensory neurons that migrate apically and extend an axon towards the olfactory bulb as they mature. In order to address whether the lack of connexin 43 in enchything cells had an impact in this process, we used controlled and conditional connexin 43 knockout mice treated with bromodioxyuridine that is incorporated to dividing cells in the basal epithelium and thus labels a cohort of new sensory neurons. As the label is retained by derived neurons throughout maturation, one can chase the label to determine a rate of maturation at different time points. In this image, you can see the BRDU label in green colocalizing with the marker of immature sensory neurons in red GAP43 at 7 days post-labeling. And here, you can see the distance that labeled cells migrated apically. Remember that new neurons are born at the base of the epithelium and they migrate up as they mature. Field circles corresponds to labeled cells that were identified as immature. We found that a week after labeling, most new neurons were still near the base of the epithelium and expressed the marker of immaturity, both in control and conditional connexin 43 knockout mice. A few days later, new cells migrate apically and lose the immaturity marker in control mice, while in connexin 43 knockout mice, the labeled cells remain in a basal location. This suggests that expression of connexin 43 by enchything cells is necessary for the terminal maturation of new sensory neurons. To address whether enchything cell connexin 43 is necessary to sustain regeneration after damage to sensory neurons, we used a model of damage to the olfactory epithelium by administration of the olfatotoxin methimazole. A few days after damage, only the basal progenitor cell layer remains in the olfactory epithelium. A week after, new immature neurons repopulate the epithelium, and a month later, the normal proportion of immature and mature sensory neurons, here labeled with OMP in green, is established. Here I show you preliminary data in which we observe the appearance of immature neurons a week after damage in control mice, but not in connexin 43 conditional knockout mice, in which immature neurons are generated later. This again suggests that new sensory neuron maturation is delayed when enchything glia lack connexin 43. We do not know yet whether this effect of the lack of connexin 43 depends on its role as a mediator of enchything cell gap junction communication or on other non-channel roles. To finish, I would like to tell you that besides our interest in the neurotrophic properties of enchything glia, we are starting to focus on another character in the olfactory nerve, which are the immune cells. Using the damage model I just mentioned, we observed that immune cells present in the terminal portion of the olfactory nerve at the level of the olfactory bulb become reactive and also proliferate after damage. This is a control with no damage. This is a few days after damage with increased numbers of immune cells, and this is during ongoing regeneration. In order to evaluate the role of immune cells during damage and repair, we plan to use genetic tools and viral vectors to express a chemogenetic actuator to inhibit the immune cells. We expect to have these new tools available in Buenos Aires in the coming months. In summary, our strategies aim at identifying mechanisms that are involved in a very efficient nervous system repair process to inspire new therapies. I would like to end by mentioning the people who did and continues doing the work, our collaborators, the institutions that provided the funding, and also by advertising the Glial Club, a community of researchers and students interested in glial physiology that meets regularly on a weekly, monthly basis. Thank you very much for your attention.
Lorena, for this wonderful talk. We are open for questions from the audience. Meanwhile, any questions appear, I may ask you something. Dear Lorena, what is the signal do you think is going on between the connexins 43 between the sheeting cells? Could it be just between them? Could be like any channels open to the surface of the cells? What are your comments about that?
Thank you, Patricia, for your invitation, for your presentation. We have a few ideas regarding that. One possibility is that establishing the network is necessary for a synchronized release of growth factors or neurotrophins or some sort of signal. The other idea that we have in mind is that connexins may work as anchor sites, mediating or facilitating migration or extension of the axons. But we will, in order to address that, we need to restore the function with, for example, connexin variants that do not have the channel function, but do have the anchoring function. That's tricky, but we are aiming in that direction.
Okay, thank you. Thank you very much. If we don't have any more questions, let's see. Well, we can continue with the final remarks at the end of the symposium. So I'm going to present the second speaker. The second speaker is Dr. Flavia Gomes from Brazil. She obtained her master's degree from the Federal University of Rio de Janeiro in Brazil, partly developed at the Weizmann Institute of Israel. Then she got a PhD in neuroscience and cellular biology at the Federal University of Rio also, with a short period at the Scripps Research Institute in the USA. Her present position is full professor at the Institute of Biomedical Science, Federal University of Rio de Janeiro. She is senior scientist of the state of Rio de Janeiro and member of the Latin America Academy of Science. And currently she is the president of the International Society for Neurochemistry. The focus of her research is to unveil the mechanisms underlying how glial cells contribute to synapse formation function in the adult and aging brain. The title of her talk is Astrocyte Functions in Aging, a Double-Edged Sword. Thank you, Flavia, for coming.
Okay, so first, I would like to thank the organizers, especially Patricia Cassina, for inviting me. It's a great pleasure to be here today, although remotely. I hope we can meet soon. So today, I'm going to talk to you about two questions my lab and my group have been working on the last decade, which are how do astrocytes contribute to the development of neural circuits, and how do astrocyte dysfunctions impact on aging and neural diseases. And in order to do that, we should discuss about aging, and we all know that the rate of the age of population has been increasing in the last decades, and we expect it to continue grow for the next following years, and especially in low-income countries. This has been associated with the increase of dementia, the incidence of dementia. Of course, this is related to a decrease in the quality of life of old people, but also can boost the costs associated with the costs of the public health. So, aging is a complex of progressive physiological changes that lead to senescence and decline of biological functions in an organism. Through a cellular and molecular point of view, aging is associated with several events and phenomena such as genomic instability, epigenetic alterations, loss of proteostasis, and several others. And my lab has been interested in two of them, which are cellular interactions and also cellular senescence. And we have been trying to understand how astrocyte aging can impact brain aging. And so in the last decade, we have shown, we and of course other groups have demonstrated that astrocyte dysfunctions are related to several neurological diseases. And we have contributed by demonstrating that astrocyte dysfunctions are related to or contribute and are associated with Alzheimer's disease and also Parkinson's disease. And in summary, we have shown very briefly, we have shown that astrocytes are present impaired properties or synaptogenic properties and neurotrophic properties on Alzheimer's disease model and also Parkinson's disease model. Today, however, I'm going to talk about how astrocyte dysfunctions can impact the health aging rather than neurological disease. And aging is associated with cognitive decline and in several species. And when we look at the molecular and cellular events related to this cognitive decline, it is known that there is decrease and impaired dendritic spine density during aging, and also alteration in neuronal excitability. And also, interestingly, associated with this, recently it has been suggested that astrocyte agent is associated with a signature molecule for the aging process. And of course, it is expected, as we know, that astrocyte can control several events of synaptic development. It is expected that astrocyte dysfunctions can impact brain aging and synapse development. However, the mechanism underlying astrocyte aging are not very well known. And in order to address this issue, so my lab has been using 3 different models. So we have been using mice, different lines of mice, and also human samples derived from elderly and middle-aged group donors. And also, as I'm going to show it to you very, very soon, we have also established an in vitro model of, astrocyte senescence. So as you probably know, aging is associated with synapse loss. So the cognitive decline is associated with synapse loss. And this has been already shown in several different species, mice, rats, and monkeys and humans. But it's interesting that besides the synapse loss It's also observed morphological alteration in astrocytes, especially from the hippocampus. So we have recently shown that astrocytes from aged mice present increased levels of GFAP and also astrocyte reactivity. But interestingly is that we also observe the presence of senescence markers in those astrocytes. And the issue about this observation is that how do we really recognize astrocyte aging? And the main problem is that senescence markers are very well recognized for several cell types, but not for astrocytes. And we have addressed this issue. So before going to our results, so senescence markers there are several senescence markers for several different cell types, and those include chromatin alteration, decreasing levels of nuclear lamin B1, and production of several different cytokines and activity of beta-galactosidase enzyme and several others. However, although some of them can be observed in astrocytes, several of them cannot. So it's a problem to recognize the difference between senescent astrocyte or a reactive astrocytes. So in order to address this issue, we have recently established a new model to study astrocyte senescence. And for that, astrocytes were maintaining for 40 days in vitro. And under this condition, those cells present several markers of senescence. So for example, increased activity of beta-galactosidase and also increased levels of nitrite. And they also produce and secrete several cytokines related to the senescent phenotype. So by using this approach, we have studied the implication of astrocyte senescence in brain aging. And so before going to the results, as I told you, there are several senescent markers for other cells. And one of these is the decreased expression of the nuclear lamin. So this protein is protein present in the nuclear membrane, and it's really important for several events such as epigenetic control, nuclear and genomic stability. And one example of a mutation in one isoform of lamin is the syndrome called the progeria syndrome, which is characterized by a premature aging. So in this condition, there is a mutation in one lamin, which is lamin A. So decreasing levels of lamin B1 is a hallmark of senescence phenotype in several cell types, but it has never been described for astrocytes. So interestingly, what we observed by using the in vitro senescent model, we have observed that astrocytes, aged astrocytes, present decreased levels of lamin B1, protein and mRNA levels. And it's interesting to know, although we don't know exactly the functional implication for this observation, we also observe that the nuclei from astrocytes present a decreased circularity, and they also present several malformations and deformations. So if we think that laminin B1 is involved in nuclear integrity, alterations in the levels of this protein might be related to proliferation and division and several other events related to nuclear function. It's interesting that all these alterations observed in our in vitro model was also we also observed in vivo in mice in the hippocampus of mice, so we observed a decreased level of laminin B1, either a general one, a global one, but also a decreased level of laminin B1 in astrocytes from mice tissue. Further, we also observed this nuclear alteration in human astrocytes from hippocampus, from human samples derived from donors, old donors and middle-aged donors. So that suggests that this decrease in laminin B1 and nuclear abnormalities are events observed either in mice as well as in human astrocytes, and maybe can be considered as an astrocyte marker for senescence. So, but the main issue is, what is the functional impact of this observation? And what is the functional impact of astrocyte agent to brain agent? So, although we don't know exactly right now, so far we don't know, we cannot answer this question. We know that laminin B1 is involved in several events. So we are not thinking that it can be only a new biomarker of astrocyte senescence, but also it can be involved in several biological functions of astrocytes. So for example, laminin B1 is involved in the formation of the nuclear lamina and also in nuclear translocation. So there are several of these events that have already been described for aged murine astrocytes. So we know that aged murine astrocytes present impaired glutamate metabolism and also impair deficit in the metabolic support and deficit in blood-brain barrier function. And also, what I'd like to call your attention is that they also present impaired cholesterol metabolism. And then I would like to call your attention that cholesterol, this lipid was one of the first molecule, the first described molecule secreted by astrocytes that control synapse formation. So we know today that, so then we decided to ask about the astrocyte synaptogenic property during aging. And just to discuss with those who maybe are not familiar with this issue, we know today that astrocyte can produce and can control several events of synapse formation, stabilization, elimination, secretion of neuroglial modulators. So they can control the metabolism of several neurotransmitters and several molecules secreted by astrocytes that can control synapse formation have already been described, and they are shown here in this table. And our group has contributed to this piece by describing that astrocytes can secrete transforming growth factor B1, and this what we have shown, to make it long story short, what we have shown is that astrocyte can control the balance between excitatory and inhibitory synapse by secreting TGF-beta1 and activating different downstream pathways. We have shown this for the cerebral cortex, but more recently, we have shown that this property of astrocyte and also this pathway might be common to astrocytes for of several different regions. For example, cerebral cortex, hippocampus, cerebellum, and midbrain. So we then decided to investigate the synaptogenic property of aged astrocytes. And to do that, we have analyzed astrocyte conditioning medium derived from aged astrocytes. And as you can see here in this part of this slide, we have shown that we have observed that aged astrocytes present decreased ability to control neuronal differentiation and survival, and also to control and to induce synapse formation. So further, we also analyze, we have also made a proteomic analysis, and we have identify a decrease in alteration in the production of several synaptogenic molecules. I'm showing here only one, which is the GF-beta-1. So we have identified that aged astrocytes from hippocampus, and they present impaired activity, synaptogenic activity, neurotrophic activity, and decreased levels of some synaptogenic molecules. So together, what our data show is that aged astrocytes from mice and also from human present decreased levels of laminin B1, and this is associated with increased outer nuclear circularity. They present an increase in the nuclear deformations, and all these alterations contribute or lead to decreased synaptogenic properties and decreased neurotrophic properties of the astrocytes. So taking this together, what we have been done from the last decade. So we have been trying to identify astrocyte pathways that can be altered in neurological diseases. And several years ago, we have shown that astrocytes derived from an Alzheimer's disease model present decreased levels of TGF-beta-1. So with this, we manipulated TGF-beta-1 pathway and TGF-beta levels by using a natural product. And what we observed is that we could rescue cognitive deficits present in aged animals and in some models of neurological disease. So taking these together, so we know today that astrocyte senescence has been associated with neurodegenerative disease, and by controlling the phenotype of astrocytes might be a very promising intervention to control this disease. And so that's what we have been trying to do in the lab in the last few years, even with the pandemic. And so I hope to have some new results to show to you very, very soon. So that I would like to thank you very much. Thank you people who made this work. We have several collaborators, but I would like to show my gratitude, especially for the blue people here: Ana Paula Araújo, Gabriela Vargas, Isabela Vivarini, Isadora Matias, Luan Diniz, and Marcelo Meloni, who really made these work. So thank you very much, and I would be happy to answer your questions if you have any. Thank you very much.
Cuento hasta 5. Well, thank you very much, Flavia, for this wonderful talk. So now we are open for questions. We have some questions from Rafael Posada-Duque that I think you answered a little bit at the end of the talk. He asked if it is possible that Alzheimer's astrocytes have increased senescent markers, and which is the mechanisms of senescent astrocyte to damage synapses. You may comment any other things on that.
Okay, so thank you, Patricia, very much for the invitation, and thank you, Rafael, for the question. So it's really very interesting too. So the first thing is that it's already known that there are some implication of decreased levels of laminin B, for example, in some neurodegenerative disease, Alzheimer's and also Parkinson, but not in astrocytes. So most of the data are from neuronal progenitors and neuronal cells. So for the astrocytes, we don't know yet. What I can tell you is that we have been evaluating the levels of laminin B1 in astrocytes from Alzheimer's disease model, and also from human samples from donors, from Alzheimer's patients. So apparently, I cannot anticipate too much for you, but what we know is that there is a decrease, a general decrease. That's something that's not new, but apparently also in astrocytes. So the functional implication, then it's a little bit more complicated. So that's what we have been trying to look for in the last, I'd say, during the pandemic, and it's real hard to know now. What I can tell you is that we know that astrocytes in Alzheimer's disease they are good and also bad, so they can help synapse, but they can also be toxic. So that's what we are working right now. I cannot tell you right now what the implications. So thank you very much for the question.
Okay, there's some more questions, but we may leave it for the end, for the final remarks, and we can go to the third speaker now, and we finally, at the end, we go on with the questions, okay? So our third speaker is Dr. Felipe Court from Chile. Sorry, I have to do an acceleration that Lorena Rela is, she made me notice that she's not a professor, she is a jefe of trabajos prácticos. Sorry, I made that mistake. So saying that, I go on with the presentation of the third speaker, Dr. Felipe Court from Chile. He has a degree in biological science at the Pontificia Universidad Católica de Chile. He obtained a PhD at the Edinburgh University with Richard Richester and Dr. Peter Brophy. He was postdoctoral fellow in the lab of Dr. Laura Fetri in Milano, Italy. Currently, he is professor and director at the Center of Integrative Biology at the Universidad Mayor, Santiago, Chile. And the focus of his research is to study the relationship between glial cells and neurons in different biological processes, including axonal degeneration, regeneration, and intracellular transfer for cargos. The title of his talk is Participation of Glial Cells in Degenerative and Regenerative Axonal Programs. So thank you, Felipe, for coming to this event.
I want to thank the organizers of this FALAN networking meeting, and especially Patricia Cassina for putting together this symposium. Today I will tell you a little bit about our work on glial cells and their effect on axonal regeneration that we do at the Center for Integrative Biology in Chile. Well, our lab is interested in local responses that take place in the axon after stimuli that are also local, and how glial cells are able to regulate these processes. This is extremely important in the axonal compartment or other compartments of the neuron that are very far from the soma, because it, of course, allows a very fast response to a local stimuli, and those are two of the main advantages of local control of the phenotype. And this is extremely important in humans, where their axons can have meters long, as well as other big mammals like the blue whale that can have axons over 30 meters. In these cases, where the axonal compartment is very far from the soma, a local response will allow a change in phenotype after some stimuli. And how glial cells contribute to this local response is what we have been interested for some time now. We have shown, for example, that axonal regeneration or axonal sprouting take place after Schwann cells are reprogrammed in a local fashion without involving other part of the neuron. We have also shown other phenotypes that are controlled by glial cells. In this case, we have shown that Schwann cells control where the node of Barbier is located. Then short Schwann cells will lead to short internodal distances, and long Schwann cells, as is shown here in this chimera, will lead to a long internodal distance that have, of course, consequences to nerve conduction velocity. And as well as in other conditions, for example, pathological conditions, Schwann cells could control where branching takes place near the neuromuscular junction. Then with this information and other information, we have published, and other groups, of course, have worked. We have came out with a model of the axon that have certain autonomy from their soma. We call it the autonomous axon, where the axon is able to synthesize proteins, have regenerative programs, and also degenerate in a local fashion. And these programs, some of them are controlled by glial cells, for example. Glia can transfer ribosomes and RNA, and we have shown that glia is able to modify the growth program of the axon, as well as regulate the initial stages of degeneration. And this is some of the problems or topics that we have been working with my lab that is shown here. Then, in terms of axonal regeneration, we know that it's highly efficient in the peripheral nervous system. This is a very clear characteristic of the peripheral nervous system in comparison to the central nervous system. And here in this scheme is shown the main step of this process. Normally, axons are associated to differentiated Schwann cells that form myelin or are non-myelin forming Schwann cells that are in this phenotype. After the axon is lost by damage, Schwann cells reprogram into a repair Schwann cell phenotype that allows a growth conformation and axonal regeneration. There's other steps also here, like macrophage infiltration, that commonly are referred to as the Valerian degeneration program. Finally, the outcome is a very efficient axonal regeneration that is associated to this reprogramming of Schwann cell into a repair phenotype. After the axon have grown, the Schwann cells can redifferentiate into this original differentiated phenotype, and as shown here, 14 days post-injury, they're able to form again myelin, okay? And the recovery of the sciatic nerve function is, in this case, in animal models, very fast. At 21 days after a complete sciatic nerve crash, there's a almost complete recovery of function. Then we are very interested to know which are the characteristics of these repair Schwann cells, okay, that are involved in their pro-regenerative capabilities in comparison to these differentiated Schwann cells that basically they don't enhance axonal regeneration, but in the contrary, they inhibit axonal regeneration. Then this repair Schwann cell is a very interesting phenotype that takes place after the Schwann cells lose their contact with the axon, both myelin-forming or non-myelin-forming Schwann cells. And the repair Schwann cell phenotype is a transitional reprogramming that is associated to a program that is very like epithelial to mesenchymal transition, where the Schwann cell increases genes associated to cell proliferation, defense response, and they decrease genes that are associated to lipid formation or myelin formation and other genes. Then this transition from the differentiated state to a repair state is controlled by two very important transcriptional factors, that is c-Jun and SOX2, when the Schwann cell lost their contact with the axon. And in fact, if you conditionally knock out c-Jun from Schwann cells, this will lead to an impairment in axonal regeneration, as shown here. That is, of course, associated to the impairment of Schwann cell reprogramming into this repair phenotype. Then these repair Schwann cells have this important function is regeneration, but also, I will go briefly here because it's already published, these repair Schwann cells are able to regulate axonal degeneration. And in fact, in the initial stages of axonal degeneration, these Schwann cells fragment the axon in small pieces with a mechanism that have the same machinery that cytokinesis during cell division. And in fact, here, when you knock down a key protein for cytokinesis in Schwann cells, that is aniline, the axon, after a damage, after separation from their soma, this axon do not get fragmented in the first stages. But repair Schwann cells also have other functions like myelin phagocytosis, increasing their adhesive properties for allowing axon to regenerate. And also something that we have been studying is how this repair Schwann cell can secrete extracellular vesicles that enhance axonal regeneration. And in fact, some time ago, we went to isolate exosomes from repair Schwann cell, and we show for the first time that these repair Schwann cell-derived exosomes were able to increase regeneration in vitro, but also in vivo, as you can see here. After nerve damage, if you inject repair Schwann cell exosomes, you have an enhancement of regeneration. These have been already demonstrated by different groups that these exosomes derived from repair Schwann cells are able effectively to enhance, importantly, axonal regeneration. Then Rodrigo Lopez and Christian Sekel in the lab wanted to know if these two conditions, okay, the repair Schwann cell and the differentiated Schwann cells in vitro were a good model to study which characteristic of the exosomes associated to this repair phenotype could be enhancing axonal regeneration. And they set up a model in vitro for having differentiated and repair Schwann cell that is shown here. This is a method that was shared by Paula Monge. Then in this model, you can have in vitro primary Schwann cells that are in a differentiated state that are expressing MVP, even though they don't have axons. c-Jun and SOX2 are downregulated. And we have also, we can have in vitro this repair Schwann cell that have low MVP, high c-Jun, and high SOX2 that is shown here in this quantification. And of course, this repair Schwann cell also have increase in proliferation. There's two methods that we use to move Schwann cells from their repair to a differentiated state in vitro. And then we ask, what's happened with the exosomes from these two different Schwann cell phenotypes? And of course, when you use exosome from repair Schwann cell in vitro, here is a DRG with the soma and the axons. This is without any treatment. These neurons don't have Schwann cells. When we put a repair Schwann cell exosome, there's an increase in regeneration, and that we have already shown. Now, but when we added differentiated exosomes into sensory axons, there's not increase in regeneration. Then basically, this transition from a differentiated to a repair phenotype also changes something in the exosomes that are secreted that enhance axonal regeneration. That this content or this characteristic of these exosomes are not shared by exosomes that are derived from differentiated Schwann cells. Then what we did was to see if c-Jun and SOX2 were involved in this, and for that we knocked down c-Jun or SOX2 from repair Schwann cells, and we isolate exosomes. And what we find was that in the case of the control, as is shown here, there's an increase in regeneration by repair derived exosomes treated with SH scramble. But when we knock down c-Jun or SOX2, they were an important and significant decrease in regeneration. Then basically, this increase in the capability of exosome for enhancing regeneration was associated to c-Jun as SOX2 expression. Then we thought maybe we can force SOX2 and c-Jun expression in cells that usually don't have pro-regenerative exosomes, like HEX cells, and for that we overexpress c-Jun or overexpress SOX2, that show here, c-Jun overexpression, SOX2 overexpression. This is our control. HEX cells don't express c-Jun or SOX2, and then we test their exosome, and in fact something very surprising, when you upregulate c-Jun or SOX2, the exosome from these HEX cells that normally do not enhance axonal regeneration become pro-regenerative. Then we thought, okay, this is a very interesting, because we can now study what are the content of these repair Schwann cell exosomes. And in fact, we did RNA-seq. We looked for mRNA and macroRNA. And then we went to see which microRNA that were contained in the exosomes have targets in important mechanisms for neuronal regeneration. And in fact, what we find is that 25% of the axonal guidance genes were controlled by two microRNA, microRNA 16 and 21, that were carried in repair Schwann cell exosomes. Then these were very interesting candidates because they were secreted by repair Schwann cells in exosomes, and they control a good proportion of genes that are associated to axonal guidance or axonal regeneration. And in fact, these microRNA 16 and 21 are highly expressed in repair Schwann cell derived exosome, but not in astrocyte derived exosome that are not pro-regenerative. And also, they are highly expressed in repair Schwann cells and exosome from repair Schwann cell compare or relative to the differentiated Schwann cell exosome as shown here by several fold in both cases. And what we did then is to produce exosomes from repair Schwann cell that were lacking this microRNA. Here is example for microRNA 21. And as shown here, when we use control exosome from repair Schwann cell, there's this increase in regeneration. But when we have exosomes from repair Schwann cell that have knockdown for microRNA 21, therefore these exosomes do not contain or have very low content of microRNA 21, we lose these pro-regenerative exosomes. When we use we knock down other microRNA that were carrying the exosomes but were not probably associated to regeneration, we don't have any effect. This is kind of a control that is somehow specific for microRNA 21. And with this, I'm very close to finish. What we are doing now is we are analyzing which happen in neurons that are treated with repair Schwann cell derived exosomes. And here is what we did is a RNA-seq comparing neurons, sensory neurons treated or not treated with repair Schwann cell exosome. And what we find, something very interesting, there were a lot of changes in these neurons. And from the top 10 GO terms, or 20, 7 of them were associated to neuronal processes that were important for or were associated to regeneration. For example, neural projection development, differentiation, development, cell projection morphogenesis, etc. Then basically, exosomes from this repair Schwann cell have strong capability to change the transcriptomic profile of the receiving neurons. And we are now studying with more details these characteristics, how they change these neurons. And with that, I want to finish. I want to thank, of course, the people that have made possible this work, people in my lab, former PhD that also participated in some of this work, of course, collaborators and the funding agencies that allow us to perform these experiments. And thank you very much, and I will be happy to take questions.
Well, okay, Felipe, thank you very much for the fantastic talk. We are waiting for some questions from the audience. Meanwhile, I would like to ask you about what do you think about the switching between the differentiated Schwann cells to the repair ones? Do the microRNAs carried by the exosome have a role also on that, or is there some signal of damage of the nerve? I don't know if you understand my question.
Yeah, sure. Yeah, thank you, Patricia. Thanks for the invitation to this symposium. Yeah, I get the question. Yeah, we are not sure about that. We have been working a little bit to try to find out if when you knock down, especially microRNAs in the Schwann cell, especially microRNA 21, we could be affecting the whole reprogramming program. We had our kind of data show that that's not the case. That will be not very good, because otherwise you will be basically knocking down microRNA 21 and changing the phenotype of the Schwann cell, and that will lead to different type of exosomes. But what we know is that microRNA 21, it controls several things related to regeneration, but also is one of the first microRNA that is upregulated after Schwann cell reprogramming from a differentiated to a repair Schwann cell phenotype. I don't know what that what means, okay, but it's a very early upregulated gene, and that at least our looking for kind of the typical markers for differentiated Schwann cells and repair Schwann cells. If you modulate it, it will not change its reprogramming. Okay, yeah. I hope I answered your question.
Okay, thank you. There's one more talk, but I can do it quickly. Daniel, we leave. There's a lot of questions. We leave it for the final, but I'm going to do one more to Felipe. Javier Ramos is asking that you have an excellent talk, and he said that so the effect of repairing exosome is pure neuronal without modulating myeloid cells, for example.
Yeah, thank you, Javier, for that question. Of course, in vitro is all neuronal, okay? But in vivo, there's several data showing that probably it regulates different things, including macrophage activation, and even exosomes could autocrine modulate, Schwann cells in terms of migration. I think, in vivo is much more complex, and there's data showing that Schwann cell-derived exosomes modulate at least macrophage and other Schwann cells. Okay, then that's I think is the more general kind of effect.
Okay, thank you, Felipe. There's a lot of questions for Lorena and Flavia and Felipe, but we leave it for the final remarks. We are moving now to the fourth speaker, who is Dr. Rafael Posada-Duque from Colombia. He is a biologist for the University of Antioquia at Medellin, Colombia. He completed his master's and PhD thesis at the same university in Medellin, Colombia. He was worked as collaborator in research with the University of Chile, the Pontificia Católica Universidad de Chile, and Johns Hopkins at the USA. His current position is professor at the Institute of Biology, Faculty of Exact and Natural Science of the University of Antioquia, Colombia. And he is researcher at the Neuroscience Group of Antioquia. The title of his talk is going to be Extracellular Vesicles from Astrocytes as Mediators of Cell-Cell Communication and Neurodegeneration Tracers. So thank you, Rafael, for accepting to be here.
Hi, everyone. Today I'm going to present the concept and different approach to study extracellular vesicles, or EVs, as cell-to-cell communication, focusing astrocytes in Alzheimer's disease. First of all, the astrocytes are cells that promote the neurovascular coupling interacting with the endothelial cells. The complex between astrocytes and endothelial is called gliovascular unit, and this regulates the metabolism and nutrient flow to the parenchyma. Although the astrocytes in healthy brain can promote synaptic plasticity, uptaking neurotransmitters such as glutamate and releasing growth factors and energy substrates, the astrocytes in disease context triggers neuroinflammatory process crucial to repair or exacerbate injury. By the way, the astrocytes mediate this process by releasing different mediators such as EVs. The EVs was initially described as just weight carriers. However, now the EVs are considered common vehicles of cell-to-cell communication and acts as a signaling activators. EVs are secreted membrane vesicles, and depending on the size, they are categorized in exosome or microvesicles. The exosomes are inward budding from the multivesicular bodies, and microvesicles are budding from the plasma membrane. The EVs can transfer different molecules such as nucleic acid, lipid, proteins, and also organelles. And these EVs can trigger detrimental effects such as A-beta deposition or increasing neuroinflammation. Regarding with this idea, currently it has been proposed the interaction between different neurovascular components can deliver EVs to circulation and to parenchyma, which trigger neuroinflammation and monocytes rolling to the parenchyma. In neurodegeneration context, the EVs has been proposed as a tracer in Alzheimer's. There are two kinds of etiology forms of the Alzheimer. The familial is associated with the autosomal dominant mutation, and sporadic Alzheimer's disease, or SAD, with a systemic alteration. SAD and FAD have different clinical and molecular alteration, and we have shown that differential EVs profile from plasma for SAD and SAD. However, it is necessary to know which is the source to generate EVs. And studying the EVs in FAD and SAD can help to understand the pathology in Alzheimer's. Based on the neuroinflammation and EVs release are implicated in neurodegeneration, and the astrocyte can generate EVs, we ask if the astrocyte and gliovascular are affected differentially in SAD and FAD. We analyze the astrocyte distribution and its morphology in human frontal cortex. The population of the GFAP positive astrocyte is limited in the upper layer of the controls samples, but spans along the whole cortex in Alzheimer's. This phenomenon was enhanced in familial Alzheimer's, which is related with astrogliosis. When we compare the morphology, as you can see here, the SAD present atrophy and FAD hypertrophy. That result suggests that SAD is related with astrodegeneration and FAD is related with astrogliosis and neuroinflammation. Then we move to the other question: is the differential state of the astroglia related with to changes in glutamate regulatory proteins in SAD and FAD? As you can see here in this panel, in green, GS, glass here green, and GFAP in red. GS is the glutamine synthetase protein, that control the excitation and inhibition processes, and GLAS control the glutamate transportation to the other cells. As you can see here in sporadic Alzheimer's disease, that patients have an increase of the GS and GLAS, and the SAD shows a decrease of the GFAP. In contrast, fat decrease that proteins and increase the GFAP surrounding the vessels. Then, in addition, we analyze the interaction between different astrocyte enzymes with the endothelial cells. As you can see here in these results, the SAD perivascular show loss of the GFAP and gain GS and GLAS, supporting our previous data. In contrast, the familiar Alzheimer's disease increased the segregation of the GFAP surrounding the vessels and decreased the GLAS in these vascular structures. All of these image was supported with the quantification. Then we wanted to know which is the changes in the vessel structure. As you can see here, the sporadic Alzheimer's disease show a strong vascular deterioration represented by gap between the endothelial cells, and also increase the fragments of the vessels along the cortex. Also, fat present or mimic the controls structure. However, that fat present increase of the thickness of the vessels. Is the GFAP enzyme surrounding associated with the alterations with Claudin localizations? Then we try to analyze the colocalization between GFAP and Claudin with lectin. As you can see here, in Alzheimer's disease, GFAP increase surrounding the vessels, and when we see these changes, decrease the lectin intensity, suggesting basal alteration. Interesting, in Alzheimer's disease, the Claudin have dot patterns or EBs-like patterns which support our previous data. In conclusions, the SAD is characterized by astroregenerations and fat with astrogliosis or inflammation. Both phenomena trigger the neurovascular uncoupling, and maybe all of these processes are involved with astrocytes changes. Then we ask if the extracellular vesicles will be involved in this damage in SAD and fat. To answer that question, we isolate human astrocytic Alzheimer's disease in cultures, and we purify EVs from astrocytes. All these EVs was used to induce activation or damage in neurovascular components. To answer that question, we propose a general methodology. We isolate human astrocytes from frontal cortex. We culture the astrocyte during 2 months, and all of the medium was collected. We isolate EVs, and then we characterize the quantity, size, and surface profile of EVs. Then the EVs was used to treat co-cultures or to induce damage in neurovascular unit. As you can see here, the human astrocytes increase increase the proliferation in Alzheimer's. Also, the fat exhibit higher confluence and fuzzy cells. Also, the familiar Alzheimer's increase the GFAP, and in that cultures, the astrocytes shed aquaporine positive EVs, which was very interesting because they support that all of the EVs confront to astrocytes. When endothelial and astrocyte co-culture were treated with astrocytic EVs from SAT and FAT, the astrocytes increased the GFAP and increased the gap between them. However, that effect was enhanced in sporadic Alzheimer's disease. Also, the neuroglial alteration was enhanced in cells treated with familiar Alzheimer's disease astrocytic EVs. Here, as you can see here, the FAT EVs decrease the branching between neurons. To support this in vitro data, the AD-EVs were injected intrahippocampal. As you can see here, the control EVs maintain the neurovascular integrity in contrast to Alzheimer's. When we compare familiar and sporadic EVs effect, the sporadic shows increase the diameter between the vessels and decrease the GS surrounding vessels. In contrast, the familiar Alzheimer's disease increase the reactivity of the GS surrounding the vessel and increase the diameter between the vessels. In summary, the AD astrocytes can shed EVs with aquaporine 4 protein, which are related with neuronal damage, astrocytic activation, and perivascular activation. And all of these effects change the vasculature structure. When we compare the SAT and FAT, the astrocytic EV generated by FAT mainly impact the astrocyte and endothelial cells, while the EVs from SAT impact more strong the astrocytic and endothelial cells. I would like to say thanks to all the family donors and MinCiencias to sponsor this research, and the master students, Julian and Juan Manuel. They performed the in vivo and human experiments, and Luis Alfonso, that is an undergraduate student implicated with the in vitro experiments.
Meanwhile, questions are coming. I would like to ask you something. It's amazing this difference you found between the astrocyte came from familiar or sporadic patients. What is your idea about that? Is that linked to any kind of mutation in the patients or any other function that is occurring in the both form of the disease?
Thank you, Patricia, for this invitation. I think we have two contexts. The sporadic Alzheimer's disease depend on their lifestyles and other comorbidities. As you know, most of these patients have a different process and systemic alteration. And we believe that the the process between the systemic extracellular vesicles impact the endothelial and the astrocytes. In another hand, the familiar Alzheimer's disease depend on the specific mutation. Most of these patients have presenilin mutations called PISA mutations, and this mutation increase the A-beta amyloid depositions. And we believe that neuroinflammation triggered by A-beta increase the proliferation of the astrocytes and also increase the GFAP reactivity. By the way, now we have a specific patient that have a resistant mutation. Inclusive in this patient have the PISA mutation. And now we know that the A-beta amyloid is not sufficient to induce the Alzheimer pathology. Actually, in this case, we have an astrocytic protection phenotype. That is very interesting. And we are now analyzing which is the impact in gliovascular unit.
Okay, thank you. There's another question, and then we go to the final remarks. Claudia Espinosa Garcia asked, that is very interesting in your lecture, and if you have considered to assess the potential role of other cells like pericytes or microglia in your neurovascular unit in vitro models.
Yes, we evaluate microglia using I/1 marker. Most of the Alzheimer's disease patients have IBA increase surrounding basals. We believe that it's related with neuroinflammatory profile. Actually, we measure interleukin-1 beta, and most of these effects are related with the IBA1 increase in there. And also pericytes, we are trying to standardize most of the markers used in the literature, but we didn't get any success with these markers. In that case, we are trying to change our methodology.
Okay, well, thank you very much. So now we can start the final remarks of the meeting, and we have a lot of questions for all of the speakers. Are we same question? Should we? So are we ready for the final remarks?
Yes.
Okay. So I have one, I'm going to make one question for each of you, and then you can ask each other. At the beginning, there was a question for Lorena Rela. Let me check. There it is. Jimena Alejandra Sierra Alta asked, when you said immune cells at the end part of your talk, do you mean microglia?
Yes, thank you for the question, Jimena. What we know so far is that they stain for IBA1. I'm sure there, as you know, the nerve has a peripheral end and has a central extension in which is quite difficult to establish what is central and what is peripheral. So I'm sure that on the central side, there is microglia in the adjacent layers superficial to the bulb. I suspect that those will stain for markers that are selective for microglia. But regarding the nerve, well, I'm reluctant to call those cells microglia because they are in the periphery, and I'm sure they will not. I mean, I suspect they will not stain for microglia selective markers, and they are more likely to be macrophages that are resident to the nerve or infiltrating from the circulation. But that's one of the questions we are addressing now.
Okay, thank you. That's very interesting because there's many work showing that monocyte, peripheral monocyte can go into the central nervous system. So this is important. So there's another question for Lorena from Felipe. that he wanted to ask you if about aging in the system that you work and what happened with coupling in that.
That's very interesting. Aging has been addressed in the olfactory system, but not with a focus on glia. So one of the remarks, one of the conclusions of this symposium is that we need to focus on glia too. It's also not addressed whether what's the dynamics of the olfactory epithelium in aging. The focus has been mainly on synapse formation and on addition of new neurons throughout adulthood. So that's one of the questions we could address, and I think the pandemic will help us because all our colonies are aged now. So we have mice in which we could address that question.
Thank you. Great. Sorry, I forgot. Then we have a lot for Flavia. There's one that Pablo Valderrama, Carmonas, that could this decrease in the TJ beta be related to the inflammatory environment during aging? Have you looked at microglia in this context?
Okay, so thank you very much for the questions. Okay, so first of all, TGF-beta is an anti-inflammatory cytokine. So of course, it helps to increase the inflammatory environment. So that's something that it's for sure. But we haven't looked at other glial cells. We all know that astrocytes and microglia, astrocyte can impact microglia activation. So this is something that certainly happens, but we haven't looked for that. But the other issue is that I think that besides the role of TGF-beta1 as an inflammatory or non-inflammatory cytokines, it also impacts synapse directly. So my feeling is that besides the inflammatory environment, the decrease in TGF-beta1 in aging is also related with synapse function rather than only to the inflammatory role as cytokines. But we haven't looked at microglia, but yeah. So thank you for the question.
Okay, there's another one from Juana Pasquini in the audience. She asked whether the senescent astrocyte another glial cells.
Sorry, I think can you repeat?
Sure. She asked whether senescent astrocytes are impacting in another glial cells.
Okay, so we haven't looked, but what do we know? So we haven't looked at other glial cells. So the question is if the senescence in astrocytes can impact, for example, microglia or
oligodendrocytes or Okay.
Thank you, Juana.
I give you another one then. So Bruno Dutra Arbo asked, he says, the cholesterol is related with the synthesis of steroid hormones. Some studies have shown that they present neuroprotective effects in Alzheimer's disease. Do you know anything about their effect in the astrocyte senescence?
No, I personally do not know, but what we know is that considering, for example, sexual hormones, there are several differences in the aging between female and male, and also in astrocytes from female and male. So it seems that they get old in a different profile. So if it's which hormone is directly involved, it's hard to know right now, but at least for the sexual hormones, there are differences if we look between gender or male and female aging, and also in astrocytes. We are looking at these right now, and it seems that astrocytes derived from male, it seems that we have some preliminary data that seems they respond to aging differently from female animals, but I can't tell much more than that, so because we don't know.
Okay, so the cholesterol is very interesting also. I'm going to go to Felipe now and then trying to see what else about this topic. So question for Felipe now. Christian Gonzalez asked, since microRNAs tend to decrease the expression of their targets and you observed increased regeneration, do you think that exosomes are releasing and break and allows intrinsic mechanism of axonal growth? Did you understand the question that I read?
Yeah, of course. Yeah, thank you, Christian, for that. Yeah, in fact, one of the ideas was that exosomes were basically releasing the break, and we went to look at the P10, that is a very important break in axonal regeneration. And in fact, what's happened in sensory neurons is that when you add a repair Schwann cell exosome, but not differentiated Schwann cell exosomes, you have a downregulation of P10, okay? That is a target of microRNA 21. And that obviously released the break of P10 that, at least in the peripheral nervous system, is very important for regeneration. And yeah, correctly, yes, that's our idea.
Rafael Posada here in the symposium asked whether it is possible that exosomes have a different effect than microvesicles in repairing neurons. There's a question that you may contribute to.
Yeah, we always tested exosomes and microvesicles, and we have like the most kind of strong effect is from exosomes, okay, or extracellular vesicles that are kind of small extracellular vesicles. Some people don't like that we continue talking about exosomes or microvesicles when they are actually a very heterogeneous population of extracellular vesicles. But big vesicles like microvesicles, they don't enhance axonal regeneration. They do other things, okay, but that are more associated to branching probably. Okay, but the strongest effect that we have is using exosome from repair Schwann cells.
Thank you.
There's another question from Marilise Murillo. She asks, do you know if some epigenetic biomarkers be involved in the extracellular vesicles, I guess?
In the effect of the extracellular vesicles, or, well, we don't know. We know that, you know, epigenetic factors or processes are involved in regeneration in the peripheral nervous system, but we have never tested if exosomes affect those kind of processes.
Okay, so I'm going to switch one question to Rafael, and Juana Pasquini asked to Rafael, how do you correlate the different extracellular vesicles in the sporadic and familiar Alzheimer's disease with the course of symptoms of the disease?
Yes, there are two studies that solve part of these questions. One of the NIH, they proposed that increase in extracellular vesicles in the periphery correlate with phospho-tau, and all of these processes in sporadic Alzheimer's disease increase with the clinical symptoms. Now we are doing some experiments, and we have the opportunity to follow these familial Alzheimer's disease patients, and we are doing a longitudinal study, and we are analyzing the quantity and different surface profiles. I'm getting some exciting results, but I would like to present this part of the results in the next FALAN.
Oh, we hope so. All of us hope. Okay, so there's another question for you, Rafael, from Eugenia Isasi. She asked, what happens with the blood-brain barrier permeability in familiar and SAD astrocyte extracellular vesicles? And what happens in other cells, like neural cells, like pericytes, or vascular smooth muscle cells?
Yes, in vitro models, we isolate the extracellular vesicles from astrocytes from plasma, and we put these extracellular vesicles in endothelial human cells co-culture with astrocytes. We were able to see that EVs increase the transendothelial flow and also increase the transendothelial resistance. We don't know why is this effect, but we are looking for mechanism for that.
Okay, thank you very much. There's still a lot of questions, but there's one for everybody who says, what's from Francisco Estrada. He says, what's the point of view of panelists about the study of astrocytes that can resolve neurodegenerative disorders? What I would like to add to there that all of the 4 speakers are presenting different phenotypes of the cells. They change from myelinated strand cells to repairing cells, or common astrocyte to senescent astrocytes. So I would like all of you to make a brief point about this change in phenotypes and what could be the clues to change one to the other one. I don't know if I was clear, but so who wants to start?
I can go. I'm not sure
Okay.
I understand the question. The question maybe is, what do we think about the potential of glial cells to function as targets for neuro repair or curing neurodegeneration? Is that the question?
Yeah, it's more or less, yes.
More or less, okay. My point of view is that it opening the eyes and adding the glial cells as a focus opens the possibilities for having new targets. I don't view them as the only solution, but they open a new set of options, and possibly those new options will interact and maybe act in synergy with the traditional options focusing on the known targets so far.
Yeah.
Felipe, yeah.
Well, I completely agree with that opinion of Lorena. Also, one thing that is important of these possible non-cell autonomous roles of glial cells in the neurons is that a lot of things that we were using or tested as therapeutics, especially in biomedical in animal models, maybe we're not targeting the neuron and they were targeting the glial cells. I think, we should kind of revisit a lot of things, taking into consideration glial cells when we do our studies. That is, 20 years ago, glial cells were, not so much considered when they were studying, or other scientists were studying pathologies or even physiology of the nervous system. And in 20 years or the last 10 years, having an explosion of new data, super amazing data about glial cells working in the physiology of the nervous system, and of course in some pathological conditions.
May I say something?
Sure.
Okay,
sure.
so I just want to, of course, I agree. I completely agree with Lorena and Felipe. I just want to add something here. I think the main issue now is to, we all recognize that glial cells, we're talking about astrocytes, are important to health and disease. But I think the main issue that we should take care is about heterogeneity of astrocytes. So one point is, I really consider that it's important to modulate astrocyte phenotypes. We know today that they are completely different. They are completely different depending on the region. They are completely different depending on the injury. And that's another issue that we should consider. So I think that manipulating astrocyte pathways and phenotypes, it's really important, but we should take care that probably it will not work, something for the cerebral cortex maybe will not work to midbrain or depending on the region. So, and also I'm gonna use this opportunity to answer, Felipe has just, he asked me about talking about modulate astrocyte phenotypes, and what about senolytic drugs, which will kill, let me say, bad astrocytes? That would be really important. But again, I think that much more important than kill bad astrocytes is to modulate their phenotypes. So that, I think that's the main issue. So maybe it took too long.
As a matter of fact, Flavia, there's two questions that are related to that. I'm going to read quickly. Javier Ramos says that, did you find altered homeostatic capacity in senescent astrocytes beside the pro-inflammatory gain of function? And Ernesto Miquel asked, once the astrocyte is in the senescent state, is there a way to rescue it, bring it back to a non-senescent phenotype? So I think these two questions are related to what you were saying.
Okay, I can briefly answer. So we have identified that those astrocytes, they have decreased synaptogenic properties, so they cannot support synapse, and also decreased neurotrophic and survival properties. And the other issue that it seems is that they also have mitochondrial metabolism dysfunctions. So that's something that we know for other cells, but rather to astrocytes. And the other question was, oh, if we can rescue
go back to the, yeah.
Yeah, that's exactly what we are trying to do now. So we are trying to rescue the phenotype, the senescent phenotype by rescuing laminin B1 levels, because we don't know what exactly induced the senescence in these astrocytes. And there are other issues that we are trying to rescue by adding in vivo and in vitro the growth factors that seems to be decreased in those animals. So that's what we are trying to do, to rescue the phenotype.
Okay, Rafael, do you want to add anything to the general comment, or yeah?
I would like to add that there is a new approach using astrocytes as a cell therapy, combining different modulators, like Flavia said. We are working now silencing some proteins related with AD pathology. And we believe maybe this approach improves part of the symptoms, but we need to improve the knowledge about the heterogeneity of the astrocytes and the specific neuroanatomical phenotypes of the astrocytes, because depending on the site, we have to change the combinatory therapies.
Okay, so we may have a few minutes more. I may go through some other question if you agree. There's one for Lorena from Pablo Hector Horacio Lopez. He said that could be possible that enchything cells communication in the regenerating olfactory nerve compensates for reduced axon-glia interaction when compared to peripheral or myelinated axons.
I don't think I followed. If they compensate,
I can copy to the chat.
Mm-hmm. It says that it could be possible that enchything cell communication in the one thing that we don't know yet, or we have only a minor idea, is what happens with coupling in the regenerating system. What I showed you is in the normal, in the healthy mouse. With regeneration, one idea that is strong in the field is that enchything cells are not changed much by regeneration. The only thing that happens is that well, they upregulate some markers that are growth promoting axon growth promoting of axons, but structurally the idea is that they remain as empty channels when the nerves die, and those empty channels are used to as a guide new channels, for new axons to grow. I don't like that idea very much. It's too stiff, or not very mechanistic, or it doesn't have the regulatory component that biological systems have. But having said that, when we studied coupling and expressional connexins in the regenerating system after damage, we didn't see large changes. So we still have to see what are the changes in coupling and in connexin expression during regeneration, and then we may know better what is their role.
Okay, so I go to Flavia again. She has a lot of questions. Many questions go around the cholesterol and lipids. For example, Eduardo Orrego asked whether exists membrane transporter as senescent marker related with no, sorry, I'm reading another one. This is my problem. Laura, here is Eduardo Orrego asked whether a specific dysregulation of lipoproteins like LDL or LDL are related with neurodegenerative effects of senescent astrocytes. And there's another one from Laura Martinez Palva that he said that you mentioned about cholesterol derived from astrocytes in synaptic function, and that is decreased in senescent cells. Is there any information about the relationship with cholesterol systemic levels and dyslipemia that is also related with age?
Yeah, there's a lot of questions about cholesterol. And so, yeah, the issue is that
it's linked also with the lipid droplets in astrocyte, perhaps. Sorry for interrupting.
Yeah, it is. But the issue is that the issue about the cholesterol was that because of cholesterol was one of the first synaptogenic molecule described, secreted by astrocytes. So it has been described by the group of Bambaris several years ago, and it has been shown to control synapse formation. And because during aging there is a decrease in cholesterol, which is secreted by astrocytes. So that was the issue. So, but I'm not sure. I don't think I can answer this question. I mean, there is nothing. There are lack of data about astrocyte aging. So I think that's the main issue of this conversation here. There are a lot of data on neuronal degeneration, but rather on astrocytes. So I don't think I can help on this issue, but yeah.
But it's important what you contribute with the observation in the nucleus and all the other organelles that we can look at. It may configure a phenotype in this astrocyte, I guess.
Yeah.
Well, I have two more for Felipe now. Pablo.
Patricia,
yes.
I'm rereading the question from Pablo, and I think I was not getting it properly. So now I understand what he asked.
Go ahead.
Yeah. So he meant that given that there is less of an interaction between axons and glia in the sheathing cells compared to other Schwann cells, for instance, in the myelinating nerve, he was asking whether coupling between sheathing cells could be compensating for the lack of those signals that axons may provide. It's possible.
Yeah.
So yeah, but we don't know. We don't have the answer.
Well, but it was nice to correct it. Just sorry, I'm reading all the questions and perhaps I don't go on time with that. So I was reading one from Pablo also, but for Felipe this time. Felipe, have you tried to testing exosomes effect using different neurotrophins for maintaining the dorsal root ganglia cells explant, other than NGF?
Yeah, that's very interesting. In fact, there's now several reports using exosomes from primary Schwann cells or differentiated Schwann cells from different different cells, kind of echoing what we find a long time, well, some years ago about exosomes increasing regeneration. And what they show, some of these data, is that they can also protect neurons from trophic factor deprivation. I think, you know, that's the closest to your question of fact that is out there. We are not working, we always use NGF. We have done a couple of very preliminary experiments taking out NGF, but yeah, they are also protective. They are not only pro-regenerative, but also they protect neurons when they are in some stresses. For example, we have clearly tested but not published because it was kind of a one-time experiment. The exosomes protect from reactive oxygen species given to the neuron. They increase kind of the protection to different stimuli. Okay. Yeah.
So, well, we have a lot of questions, but I don't know if we have time to go ahead. I don't know if you want to do some very little notes to round out this conversation.
I would like to say that these events are very important. Sometimes we are much closer than with other groups that work in countries in Europe or in North America, and we know more about the work of others than we know about people with whom we could collaborate much more easily with less expense. And of course, we don't have as many groups that do good science, but we have many that do. Yeah. So we could many times find the tools and the interactions closer than we might think.
Well, thank you, Lorena, for that saying. In the organization committee, we are very careful to select only Latin American speakers, trying to be equally in gender and all these things. So we are glad that we finally could do this virtual meeting, and in preparation for the next one, who is going to be, hope so, next year in Belén. So this is nice word, and thank you. Any other one who wants to say anything?
Yeah, Patricia, I just want to, again, to congratulate you all, because we have been experiencing a very, very hard time, a very, very difficult time during the last 1.5 years and 2 years. So it was really nice to meet you all, and especially to discuss about that issue that has been growing a lot in the last few years, which are glial cells, and that's exactly what Lorena just said. So then we can increase and support collaboration between South American groups, and that's really nice. So thank you very much and congratulations. And also about the answer, so maybe we can send to you, to the organizers, the answers for the questions that we did not have time to respond. Sure. So
in fact, we are going to make a forum in the platform about each of the events, and we can continue there. So I'm going to copy all the questions there. The presentation are going to be online for some time. We don't know yet how long, but they're going to be there. So, and at the same moment, it's going another symposium that might be interesting for us, which is in neurodegeneration. So you can see the other symposium just in case to increase the inputs for our central nervous system. Okay.
Yeah, I want to say, I think Lorena and Flavia probably said the most important things that I think that's kind of the core or the crucial thing that we should kind of work together, and these networking meetings and other meetings that have passed in the past pre-pandemic have also kind of tended toward that, to make groups in South America, Latin America, and the Caribbean. And yeah, I hope that we see each other soon next year, okay, for the FALAN. And thank you very much for the organizers, and you, Patricia. Has been great to be part of this and see such familiar faces after a lot of
so long, after so long.
Yeah, it's great.
Well, thank you, Rafael. Do you want to say anything else, or just?
Yes.
Yeah.
I would like to say thanks for everyone to all these beautiful presentations. I think this inspired me to to do many things and also to get collaborations. And I would like to say thanks for all the people asked through the chat, because this topic is very interesting for the neuroscientists. And we believe that we are a good approach in Latin America to increase the knowledge about the astrocytes and glial cells.
Okay, thank you very much. So I thanks again for any of you to come here. And I remind you that we have another activity in the afternoon. We are going to set our code of conduct. So it's a very important political activity for the FALAN. So thank you again and see you next year, I hope at Belén or whatever. Okay. Well, thank you very much.
Gracias, Patricia.
Bye.
Muchas gracias.
Thank
Bye.
you. Gracias.
Bye.
Un placer,
Gracias.
un placer. Chao, chao.
Buenas tardes a todos. Es un honor estar acá nuevamente. Después empiezo a hablar inglés. Nunca sé qué lengua hablar cuando estoy hablando de FALAN. Good afternoon. It's a great honor for me to be here with you. And today we are talking about things related to science. First, we are going to discuss in a minute the new code of conduct in FALAN. But who is going to speak about that is the Professor Ana Silva. She's a professor at the University of La República in Montevideo, Uruguay. También es miembro de LARC-IBRO, de Comité LARC-IBRO. Entonces, sorry, I will pay attention. In a minute, in some minutes, she's coming, okay? But first of all, I would like to discuss some aspects concerning FALAN role. As I said yesterday, FALAN has a mission. What is that mission? It's to increase the interaction and exchange in Latin America, in and outside Latin America. Increase or promote education, training, neuroscience. Also to that the decision makers and the general public, that they can understand and be involved in neuroscience. And also to promote discussion in animals research, women's rights, and science birth. Okay, I just want to show you some aspects concerning women's rights and science financial burden that FALAN has been promoting the last 2 years, okay? Just because Osvaldo Chitel, Professor Uchitel, talk about Professor John Nichols, I would like to show him here. It was the first time that he came to Ribeirão Preto. It was an IBRO VLTP course. You can see here Norberto Cairasco, he's from Colombia. Marcela was my former student. She's from Colombia also. And you can see here Bill Adams, Kenny Muller, and there is one more that I don't know. Oh, Francisco Fernandez de Miguel, de Mexico. I'm sure if you look, maybe if you really look, you would see many people that you know here. It was a marvelous course, okay? So the first question that we have is, how can we improve FALAN? No, that is the main, what we need, we are going to talk today, no? So FALAN did, and I hope all you will pay attention on it and fill up the forms in order to have the Latin American map of neuroscience, okay? We need to know the researches in Latin America, the subjects of study, the techniques, and it's a way to enlighten Latin America and Caribbean to have more collaboration between us. We need that, okay? We need to increase communication. Daniel and Sue team help us, and we have a website. We have our information in Facebook and Twitter. But what else can we do? What else FALAN can do to communicate with the other ones in Latin America? This was the second course in Paraguay. You can see here, John Nichols also, Professor Uchitel that was there yesterday, 3 Brazilians, former students of John Nichols, and Professor McMahon that was the boss of IBRO VLTP. Another thing that we need to do is increase FALAN community, more countries. And you see today in the assembly that we have more candidates to be part of FALAN. It is important also. And now it's more than time to talk about ethics, gender, and minorities in science, okay? That's something we didn't talk a lot some time ago, but now it's time. Okay, so the first thing is to think about women and underrepresented minorities in Caribbean and America Latina. We have a lot, that's true. And despite we don't know, we are all biased. We need to pay attention, okay? Women and minorities, they do not receive equal treatment in the activities for progression of the academy later. And there is no difference of this behavior, of this treatment in our countries and in the USA or Europe. We have several publications that show it. But in the Latin America, we have more prejudice, race, socioeconomic status. This is something that we need to think about. And it is absolutely necessary to have or to have for them impartial access to education and research. We have a lot of difference here. And the COVID-19 showed this in a deep way, okay? The inequalities are stronger than ever. So, and even us that we, let's say, have a position in the university or in the institute, we are suffering because in our countries, there are much less science investment. We need to bring it to discussion in the neuroscience societies, and FALAN can integrate that. No, what can we do? The first thing is that we shouldn't deny that those things are happening. We need to be aware of the implicit biases in our world. We need to do that, okay? The other thing, the problem with gender is not just because gender. It is because we, women, need to work much more. We needed to make a big effort over the course of entire academic career. So many women just stop to work. They cannot allow themselves to go to such a difficult time. And it happens, it is true, okay? And we need to pay attention, no? That it is important to invite representation as speakers at meetings, members of committee and positions of journal leadership. I remember Patricia Cassina, she was telling, I'm very glad with this meeting because we have a balance between women and men. So FALAN is trying to accomplish this also, okay? It is important for all of them to have safety to conduce work, to obtain research support. That's something most of the ones in the committees are men. They need to think about if they have any bias or not. And also, not just us, but in all over the world, it is important to increase the number of mentoring women underrepresented minorities. And also increase citations. We must trust the work done. Why? Because marginalized groups are under citation. And we know that. It's easy for us to see the impact of a manuscript, a good one, of us from below the equator. It's a difficult situation. I don't have, FALAN doesn't have a recipe to go over that, but how we can, what can we do to make it better? And all the work we do, we need to publish. It is important, okay? I found out that there is a Women in Neuroscience repository. Here is the link. And in this link, there is a list of neuroscientists all over the world. And it's possible to identify and recommend female neuroscientists for conference, symposia, or collaboration. Please, if you don't, you are not part of this Women in Neuroscience repository, try to do that. Just go to the site and put your name there and what you do. I know maybe in a near future, Danielle, Paola, Patricia, we can do something like that in our platform, no? Why not? Okay, so another thing that this is my particular worry, and I have been talking with people from Europe, is the open science publication. If I don't like to have all the articles as an open science, I love it. If it's very nice, don't need to pay to have the journal. It's perfect. But in my point of view, despite all the good things that it brings all one more expense for the research. So now we need to pay for the fellowship for the students. We need to pay for the lab, everything that we have in the lab. So we need to have good grants. And now we need to pay for publishing our results. Okay, it is important. But for example, in my lab, we don't have 2,500 euros to pay for just one publication. Okay. So what I do is there are many journals that are hybrid, and there are ones that are just, you just can publishing open science, but very, very expensive, impossible to do that. And there are ones that are really completely, you don't need to pay for that, but you pay for the editor, no? So less well-funded authors and authors from less developed countries would be less likely to submit an open science publication. It's necessary to think about a system that is open in an equitable, and equitable for all. How? What's to be done for our community? More global investment in education, research publishing capacity. Maybe to have meetings with World Bank, the World Economic Forum, UNESCO, and other actors in this space. We needed to have a position. It's difficult just, I have money, I'm gonna pay for my publication and so on. And trust me, people from Europe, Max Planck Institute, or Switzerland or US, they don't know about that. They think that everything is easy for all of us, okay? And another thing that I learned was about the brain drain. I found this comment for Thorsten Wiesel, that was a former president of IBRO in 2014, okay? And as he said, it is important for, if you want to become a great scientist, you need to be exposed to greatness and another language also. So, it is important not to have all the young people that doesn't have a job in our countries just go and go for this period and don't come back. So it's necessary to have also overseas scholarships that encourage scientists to return home. IBRO has got this return home fellowship, but it's not enough for all of us. I don't want to talk too much. So the last thing is that FALAN signed last year the ALBA Declaration. Anna is going to talk about a little bit what is the ALBA Declaration, what is ALBA, and this declaration and other things that happen in several societies inspired us as FALAN to have a code, to elaborate a code of conduct. She's going to present it for all of you today, and also this code is going to be voted if it's approved or not in the FALAN assembly later on, okay? But for the ones that I would like to leave a message to take, oh, this is Anna, she's coming in 1 minute, but I would like you to take home this message. This is the Prime Minister, Maya Amor from Barbados, and she gave a talk, 15 minutes or 16 minutes, not too big, but it was at the United Nations last week. And what she said, it was amazing. If you have time and just have a look or listen to, it's very nice. It's very nice. So now I'm gonna let you with Ana Silva. And in the end of this talk, we have time for questions, for discussion, and all of you are really welcome. It's a pleasure to talk with all of you, okay? So see you later. Anna, it's your time now.
Thank you, Elaine.
It's okay. I think you can share your. No?
Right. Let me see. Okay, am I sharing my screen well? Great. Great. Okay, thank you very much, Elaine, for your introduction and the very interesting things you were mentioning before my talk. Good afternoon to you all. It is really a big pleasure and honor to be here today. I'm so glad to have the chance of interacting with our Latin American colleagues after such a long time. And as representative of the Sociedad de Neurociencias de Uruguay, and also as IBRO LARC member, as Elaine mentioned. I would like to present today what was the history and the progress of this initiative to have a code of conduct in FALAN. So this code of conduct that will be probably and hopefully approved today in the assembly that we are going to have after this session. And I asked Danielle to send us in the chat box the link for you all to see these, the last version that we have written about the code of conduct. So you can be able to check it during this talk and the discussion after it. So when we think of a code of conduct, the first thing that appears to me is why do we have to have a code of conduct? And the response or the answers to these questions are very different, but for us, for FALAN, it is clear, it has become clear, very clear, that diversity is essential to delivering excellence in science. There is a global agreement in that. So a diverse and inclusive scientific workforce draws for a wide range of backgrounds, perspectives, and experiences. And this, of course, improves innovation, creativity in science. So diversity, we do think now that the diversity facilitates specialization. It also invigorates problem solving. It also balances these biases that Elaine was talking about. But without strong policies of equity and inclusion, diversity cannot be maintained. So that is something that we need to be very careful and pay attention to. For us, for Latin American scientists, we all know that it is more difficult to work in science in Latin America than in the other parts of the world. It is more difficult for us to be part of the international community of science in which we are clearly underrepresented, and we for long feel this not belong to this community. But to invigorate this or with this purpose, not only with the purpose of belonging or to try to be more represented, it is also true that Latin American neuroscience community, us, we are also a very diverse community. And within us, we have to be very careful about our own diversity and to make our communities a safe and welcoming place for everyone. So this is part of why are we talking about a code of conduct in FALAN. But it is also important to mention why are we talking about this now? And the first and most important thing that I think that was very inspiring for us was what Elaine was mentioning about the ALBA network. As you probably know, the ALBA network is a global initiative that aims to promote equity and diversity in brain sciences. And this network has launched a declaration on equity and inclusion that you can check in this link here in January 2021. And I think this is an important landmark because this declaration is the first of its kind, to my knowledge, to propose not only to state or to make a statement or a declaration on what things are good or wrong, but it also provides positive evidence-based actions that individuals and organizations at any level can take to promote equity and inclusion. So this declaration goes beyond the statements and promotes actions to be taken. to somehow counteract implicit bias and workplace culture. Until today, more than 200 institutions across the world have signed the ALBA Network Declaration. And of course, FALAN, and not only FALAN, most, I think, of the national neuroscience societies from Latin America are among these institutions that signed this declaration very early at the moment it was launched. And one of the actions of this ALBA declaration is to adopt and enforce a code of conduct. Oh. So this is one important thing. We are just like, we signed and approved, endorsed the ALBA Declaration Network, and we are now taking one of the actions that was promoted by this declaration, to have a code of conduct. The other two important things that make this this moment a good opportunity to have this code of conduct for FALAN is, of course, that we are having this FALAN network meeting this week. And this was a great opportunity to have this code of conduct discussed and approved. And of course, in third place, that it is also timely for the organization of our next meeting in September 2022, in which we are for sure going to meet again in person in Belém, Brazil. So at this moment, what is it? What is a code of conduct? Code of conduct is kind of, in this code of conduct, FALAN will declare the behavioral standards to maintain during the congresses and events organized by FALAN. We'll also review how to receive claims of misconduct and also the ways this misconduct will be sanctioned. And it is important to know that this code of conduct is that it's exclusive and restricted to FALAN organized events. And what the purposes and the aims are to ensure an inclusive and welcoming environment for all participants. I like to think this as a private party, you know, the organizers of FALAN or events are organized by FALAN and are private events in which organizers set the rules. And these events are planned very carefully, with lots of plans, programs, speakers, topics to be discussed. And there's a lot of effort, a lot of thinking, and these events have to turn out right. And so, as the same example, I like the example that I heard in one of these meetings talking about this code of conduct, that is an example of a big family party, or for example, the Fiesta de 15, the 15 years old ball, in which all the family has put a lot of effort, a lot of money, a lot of thinking of details. And of course, this family and these organizers wouldn't like to have it ruined because one person is misbehaving. And of course, if that happened, they will have the right to say to that person that it does not belong to this place and it should go and leave. So the FALAN Code of Conduct is a way of setting norms and boundaries in how to behave in these events organized by FALAN. And it has been inspired in the code of conducts of other societies in its present form, we worked on it based on the Society for Neuroscience Code of Conduct, and also based on other international societies in neuroscience. And this Code of Conduct, specifically the FALAN Code of Conduct, has the same structure that most Code of Conducts have. And this means that they contain a short statement of the goals of this society, in this case of FALAN, of having this Code of Conduct, then a list of unacceptable behaviors that cannot be observed in FALAN events, description of where this Code of Conduct applies, very clear. In this case, it will be only for FALAN organized events. And then simple instructions on how to report violations or misconducts with respect to this list that has been addressed, and the consequences of violating this Code of Conduct. So this is what we worked on in this past 2 months, and this is what we are going to, or we are proposing here today to be voted in the Assembly, and before that be discussed in this session. It is also important to say what the Code of Conduct is not. So the Code of Conduct, it is not about punishing people. It is clearly not about repairing the harm caused to potential victims of bad behavior. And also, it is not about smoothing over conflict or mediating between people. It's a statement of what things are expected and what things are not expected to happen in a healthy, welcoming, and safe event. As you see here, these are the actions that have been taken so far to be here today discussing this FALAN Code of Conduct. In May, we have this idea, and it was the Sociedad de Neurociencias de Uruguay who proposed the FALAN Directive Council to work towards a Code of Conduct in FALAN. This idea was received very enthusiastically, and we immediately began to work on it. And so we worked with Elaine as a subcommission of writing this Code of Conduct, and we have a draft ready at the beginning of August. This draft was, this version was distributed to all the FALAN members, all the societies, national societies, and we received feedback from some of them and suggestions and editing of some of the parts. And that was actually done until probably last week or last weekend, maybe. And we are now ready to put into consideration of this FALAN Assembly. The final, or maybe the last version is the strict way to call it, of this Code of Conduct for its approval. And so, as I told you, you can be checking at this moment. The Code of Conduct of FALAN is a 2-page document. It's short, it's clear, and I have in the 3 next slides, I have selected the main parts of its last version. And this is not the complete version, but for you to have an idea, I won't read this, but this is the first part in which the Code of Conduct states its aims and states the purposes of having a Code of Conduct and where and this Code of Conduct will be applied. So, and it also says that all FALAN members or participants in FALAN events shall be familiar or know about this code of conduct and have probably need to endorse it before registering in one FALAN event in the future. The second part is the way code of conducts used to declare what we understand by misbehaviors in a FALAN event. And these misbehaviors are clearly and stated and listed. So we have sexual harassment, discrimination, bullying. And if you read with carefully, there are different examples to address clearly what we mean by these misconducts. And finally, in the last version and last part of this code of conduct, we do have the way this potential misbehaviors during FALAN congresses or events have to be reported. And what happens when these reports reach the authorities of FALAN? So you can read this carefully, and you may have several questions or even suggestions to change. The code of conduct is never a final version. It can always be modified and amended as people find or suggest things that are missing or things that are not adequate to the society it is about. So just finishing, what is next now? Of course, after approving this code of conduct, after having had this chance of stating that this issue is important for FALAN and that FALAN is going to be paying attention to these issues in this way, it is important to enforce the FALAN code of conduct, and all the activities after its declaration will be like focused on its enforcement. What does enforcement of a code of conduct mean? Well, it is probably, we will probably need a code of conduct or a diversity committee. This committee should be, will be dealing with adapting this code of conduct to the real situations that appear in our FALAN, and also will need to be trained in how to deal with this report of misconduct, how to evaluate them, how to be fast and have a good protocol to evaluate this, and how to have a real presence in these future FALAN events. And the enforcement of FALAN, of course, will also mean in the next steps to include the endorsement of the FALAN Code of Conduct in the registration process of FALAN meetings. Similarly, as we do when we are filling the form of any form to register to a Congress, and it probably happened to you all if you are registering to another meetings at this moment. At one point, you have to read and endorse the code of conduct. So all the participants of a FALAN event in the future will know what is the behavior expected from them in these events. So I'm finishing here. I really thank you very much for your attention. As I mentioned, we are looking forward to the discussion and questions you might have today. And of course, totally open to suggestions, inclusions, and deletions of the code of conduct in the version we are going to discuss it today and approve it in the FALAN Assembly. Thank you very much. Okay, Elaine.
Thank you very much, Anna, for your nice presentation. She was very clear. We needed to have something like that. They just informed that there is no questions in the chat. But I was wondering if I could ask you a chat, and maybe you ask me once, so we keep talking for a while. Okay. This code of conduct, for sure, it is written also the things concerning women, diversity, and prejudice, and everything, no. And I was a little, let's say, I was worried because in the beginning I thought, no, God, it is going to give law to us, not law, but you know, something like, and it's going to be difficult. Everybody's so happy in our meetings, no? I didn't have any idea that would be necessary to have this kind of code. But maybe you told me you did a course, may I see, about with lawyers and things like that. Maybe you could just give some examples of not good things that happens eventually in meetings, and this code can help to avoid those things. No, it's the idea. No, yeah.
Yes, one important thing that it is being like establishing by the recommendation of the ALBA Declaration, and also what all the societies really worldwide are doing, is not thinking about this code of conduct as something that is going to be sanctioning people, like punishing of misconduct. It is just quite the opposite way of thinking about this. It is a declaration of how we expect people to behave. And this is to be welcoming. It's not going to, okay, I'm going to be watching you if you are not good or doing something wrong. It is exactly to protect all the attendees to an event of having clear ways and expectable ways of behaving. And I also think that this is important to think about in this way. As you told, people are, I think that us, from our generation, we always neglect that things like this happen. And they, of course, happen in several ways. Just a person that has a title or is a professor, and the way he or she might be joking about some other person in a meeting can be offensive for other persons. So this is not only about sexual harassment or discrimination, of big, huge problems on this. This can also happen and be educative for all of us to how we have to address and how we have to be responsible and conscious of the status we have when we are in this kind of meetings. So here we have questions.
Yeah.
There's one question, I don't know who is Lapnek UFSC. Why do you think?
It's from Santa Catarina.
Santa
University
Catarina.
Federal of Santa Catarina, yes. Okay, I think it's for you, yes.
Okay, is why do you think this code is necessary? What is this improving? Maybe I was talking a bit about that in the last minutes. What we are doing with this is first addressing that we know that these things happen in events, and we want people to know that we are being alert of them so they can come with confidence, they can be calm, because people are not going to be we are going to be like trying not things to happen. So student from, I don't know, from any university that is unsecure of being presenting his poster will know by this ways of stating clearly how to behave, that he or she would be asked politely about their results in a poster, will not be biased or will not mention any bias about their aspect, their sexual identity, or the ethnic origin. So if someone that is worried about that, this is a welcoming place for them. And we are saying this out loud. We are putting this or written in a declaration.
There is another one from Francesco Rossi.
Francesco, I suppose we need an ethical committee supporting the directive committee of FALAN. Yes, I mentioned this in the presentation, in the slide, that what's next. And of course, the next thing to do, because if this is important, the FALAN directive committee has to endorse. We cannot do this and only have it like a document we have signed and we will not do anything else about. So if this is important, we have to have a committee. We have to be always remembering all our membership, all the attendees of an event, that we are paying attention to this. And if there is something that is going out of this predicted or recommended behaviors, the society, in this case, the Directive Council of FALAN, will be taking actions. So actions have to be taken if things go out of normal, and the way to take actions needs to be trained. And so these committees need to be populated by people that somehow are ready to deal with this type of situations. Hopefully there will be no situations, and that's great, but I'm afraid that there will be at least a couple, maybe minor, or things that wouldn't mean any strong action. But it is important to talk about this and to have this committee to deal with this potential misconduct reports.
Yeah. Just a comment, no? The implementation is the fact, something next to the declaration. Yes.
Exactly.
Yes. Well, it's something for the new, we hope, no, the new committee that is coming. Okay.
Yeah.
Okay. There is no more questions. I don't know if you would like to comment something, Anna. No.
I think I talked a lot.
Okay. You talk a lot. No. Thank you for coming, for doing the hard work and to present it to all of us. I think, Daniel, we have some time until the FALAN assembly, no? It is 3:30, no? If I'm not wrong. So now we should, thank you. Okay, so we will have a short time to rest, and so after that, one more meeting for the societies and the committee. Thank you very much. I believe for the most of you, that is the end of the meeting today. But don't forget, we are going to be here tomorrow. And as I said in the first day, let's share our passion for neuroscience. And as I read many comments in the chat, for sure, Latin America and Caribbean make wonderful science. Okay, thank you very much for today, and I hope to see you with us tomorrow. Good afternoon.